Okay. Okay. So, thank you for this welcome. So the flow for this morning, we will talk about the organization for the day later in the morning because some people is arriving during the morning. So maybe in the afternoon we will talk about the organization. You see, everyone has his own desk. We can work and participate in the discussions. The flow is going. We are going to have an invited talk first, and then short time for questions and discussions, and then we start with the talks in the presentation. We had to change some of the presentations because there is some people that is arriving late today because some flights problems, and we have to change a little bit the program. And I think now is the time for the first talk. As you know, this kind of meetings is I am very happy of being part of this group, and the main thing is that no question is without an answer. I mean, I ask to you to do good questions, to favor the discussion, and don't left any question in your brain. Just go out and make the question. And we have time enough for discussion, so that is the point of this kind of meetings. I'm going to introduce you. Juan Antonio is a professor of forest pathology in University of Madrid, Polytechnical University of Madrid, and he's a good friend, and as well he's a very good scientist. He's in charge of the the Dutch elm disease, and he's going to present the program of it's a long time program, but they are doing very nice things. Come on.
Hello?
Yeah.
Yeah? Okay. Thank you, Luis, and thank you, the organizing committee, for inviting me to this nice conference. It's an honor for me to open this meeting. I'm going to talk about breeding in Spain against Dutch elm disease. Okay, first of all, I'm going to briefly introduce Dutch elm disease. Dutch elm disease is a vascular disease caused by two fungi, Ophiostoma ulmi and Ophiostoma novo-ulmi, which develops inside xylem vessels, causing their embolisms and occlusion. The tree develops a wilt syndrome, and the disease is mainly spread by bark beetles of the genus Scolytus and Ilurgopinus. There have been two big pandemics, two destructive Dutch elm disease pandemics. The first one was caused by Ophiostoma ulmi at the beginning of the past century, which was moderately aggressive to European elms and quite aggressive also to American elms, but less than European elms. Second pandemic was caused by Ophiostoma novo-ulmi, which replaced the first species. And this species was highly aggressive to both European and American elms. The situation that we have today is that adult elms have disappeared from the landscape, but we now are in a post-epidemic phase with recurrent cycles of high disease incidence in recruitment trees, as can be seen in this photo. The Spanish breeding program started in 1986, was founded by Professor Luis Gil. In this photo, we can see other European colleagues in a project about elm conservation in Europe. Other key person was Professor Alejandro Soya and Margarita Buron at the beginning of the program. The Spanish program took as a reference other programs in Europe, such as the Italian program and program in the Netherlands, and also programs in North America. Main goals of the Spanish cell-breeding programs were to understand the history and variability of the remaining populations, to conserve the remaining genetic resources of Spanish elms. Today we have more than 700 genotypes conserved in ex situ conservation plots. Identify resistant mechanisms of elms, and to obtain resistant elms by means of traditional breeding. I joined a Spanish breeding program in the year 2002, and my main focus was this objective, to study resistant mechanisms of elms, so my talk will focus on this aspect. The first breeding efforts took place in the 90s and was made by means of artificial crossings between our most abundant native elm, Ulmus minor, and Asian elms, Ulmus pumila, which is quite resistant to the disease. Through this strategy, several resistant varieties were produced, and several findings, important findings from a research perspective, were that resistance was mainly transmitted from the mother. For instance, if the more resistant hybrid minor by pumila acted as a male parental tree, the wilting was higher than if this same hybrid acted as the female parental tree. High degree of additive genetic control of resistance was found, supporting breeding, and resistance was related to early bud burst of trees, as was also observed by Italian breeders before. Professor Soya, in the year 2000, in his PhD thesis, stated that resistant level of Spanish population, the native population of Ulmus minor, is low, but selection of resistant individuals is feasible. In fact, he preselected in his thesis 14 Ulmus minor trees to be part of the breeding program. And he also started to study resistant mechanisms of elms. No? We observed that elms with large vessels tend to be more susceptible to the disease, mainly because large vessels are more prone to embolisms and favor pathogen dispersal. Here we can see what can be a susceptible tree and a resistant tree. And if we assume that the larger vessels are the first lost by the disease because of embolism or occlusion, then the resistant tree will have more vessels of lower size, which are able to sustain water flow during disease. No? We found that this relation is true at the beginning of the season, coinciding with the peak of bark beetle flight. Later on in the season, however, this relation is reversed. So Ulmus pumila, the resistant Ulmus pumila, show larger vessels than Ulmus minor. This means that seasonal changes in wood formation should be taken into account when comparing vessel size in relation with resistance. In the 2000s, hybrids with pumila were progressively eliminated from breeding cycles, mainly due to conservation concerns, and because breeding with native material was considered as feasible. And we progressed in the study of resistant mechanisms of elms against the Dutch elm disease, and we found that not only vessel size matters. There are other traits that can be also related with resistance. For instance, intervessel pits tend to be larger, also in susceptible than in resistant trees, probably favoring a spread or toxins from the pathogen or other processes. We also found that in susceptible trees, starch is depleted at higher rates than in resistant individuals. Here we can see hyphae of Ophiostoma novo-ulmi penetrating ray parenchyma cells, which are rich on starch grains. And probably more important, we found that common response of elms against the pathogen is the formation of tangential bands of suberin-like compounds, forming what is called a barrier zone. These barrier zones separates infected xylem from healthy one and protects the cambial area. we've also found that the resistant trees tend to form thicker bands of barrier thorns than susceptible trees. An important finding in the conservation program was the characterization of European Ulmus minor and also Ulmus glabra population, and the finding that a high proportion of our elms in Spain were clonal, was a single genotype. This genotype corresponding to the Athenian elm, an elm propagated by Romans in the past, and is widespread in Spain and England. And this, of course, had important consequences for the breeding program in terms of conservation and in terms of breeding. As we had previously, we had many replicates of the same genotype conserved in our clonal banks. The first resistant Ulmus minor trees were registered in 2014. These trees are not immune to the disease. They show about 30% of foliage wilting when they are inoculated with the pathogen. Two of them, however, were subsequently we found that two of them were introgressed with Ulmus pumila genes, so they were not used anymore for planting in natural environments. A live program, Live Elm, supported the plantation of these resistant genotypes mainly in riparian areas in central Spain, and also in urban green areas, recovering a traditional use of elms as important urban trees. The program was important also because it reached an important social impact and a great replicability. After the project, more than 40,000 elms have been planted throughout Spain in 38 provinces in different environments. In some cases, occupying emblematic places like this one that were previously iconic large elms were present. No? In Spain, it was a tree very close to the tradition, and it was quite often that a big elm was planted in the square of the town. By now, plantations are in good health. Occasionally, we have detected some symptoms in some of these varieties, more or less the same as we found in our inoculation trials. No mortality by now, and two of them haven't shown any death symptoms by now. On the other hand, in 2010, we obtained two crosses of resistant by susceptible trees. This is a way to preserve diversity from susceptible individuals. No? We obtained two families that were clonally propagated and planted in an experimental plot that was inoculated with the pathogen. And here we can see the behavior in terms of foliage wilting of these two families. We observe how the mean wilting value of the two families was closer to the dam progenitor than to the sire, the more resistant, supporting again that resistance is mainly transmitted from the mother. Furthermore, we observe that some genotypes, 2.5 of them, behave better than the resistant progenitor. Two of these clones are now in process of registration as new resistant varieties. This is, of course, a way to increase the genetic pool of resistant individuals by crossing with susceptible trees. But it's, of course, a lot of effort in terms of plant propagation and inoculation. In the same two families, we investigated what are the causes behind the resistance of the most resistant individuals, no? And we found that in the first family, the more resistant individuals, those showing less than 50% wilting, are associated with wide and few early wood vessels. This, in principle, is surprising because resistance is normally associated with small vessels, but in this case, we observe that the more resistant individuals tend to have wider vessels, but in lower number. This strategy could be associated to a water use efficiency. So tree seems to invest in trying to conduct water better, no? While in the other family, the more resistant individuals were associated to narrow early wood vessels and more frequent vessel occlusion, and a strategy that could be more related to xylem safety. This evidence that resistant to mutagenesis has a multifactorial nature, is not only one type of resistance, and that diversity of resistant mechanisms exists among different genotypes. And this is good from a breeding perspective. Sorry, se ha vuelto loco. In 2014, an important milestone, of the program was the development of in vitro propagation of elms, which increased our capacity to propagate plants a lot. And also we developed an in vitro early screening method, which is very important because conventional testing takes around 7 years, because elms should be inoculated in the 4th year to show symptoms. But with this method, we inoculate small plantlets through the root system in vitro, and then we measure some basic biochemical parameters and also plant growth, and it's able to discriminate quite well resistant from susceptible trees. So we are incorporating now this technique to our breeding process. I want also to highlight work of my PhD student, Clara Martinez Arias. She compared the behavior of one susceptible with 2 resistant clones, Amaniel and Ademuth, in terms of upregulation or downregulation of genes related with resistance, some associated with the salicylic acid pathway or the jasmonate ethylene pathway. And she found that the susceptible and the Ademuth resistant tree show quite high response, but the Ademuth showed this response earlier than the susceptible tree. On the other hand, Amaniel showed quite mild response to the disease, lower overexpression of genes. Examining the xylem characteristics, we found that the susceptible tree had longer and wider vessels than the 2 resistant trees. And Amaniel has especially low vessel diameter, small vessel diameter. This is probably the cause because this genotype has lower presence of the pathogen in their tissues. This finding, together with other studies, led us to conclude that these 2 resistant clones show quite distinct resistant mechanisms. While Amaniel relies its resistance in having narrow and short vessel occlusion, and an early antioxidant response, Ademuth relies its resistance in intense suberin barrier thorns, as can be seen in this photo, and a strong and early salicylic acid-related response. So if we could combine these 2 layers of defense in a single genotype by means of artificial crossings, we will probably obtain a genotype with superior resistance. We are also investigating the role of the endophytic microbiome in resistance. We found, we first defined which can be called the core microbiome of elms. We found that some yeast are widely spread through the crown of the tree and are quite present in different locations. And 2 families of yeast seems to be quite related with the resistance of their host tree. No? We are now also investigating the effect of these endophytes in plant performance, and we found that some in vitro plants inoculated with the pathogen show a higher survival than control plants, against abiotic stress. And by inoculating also trees, we found that some endophytes are able to increase resistance during one season, during the first season. Later on, this effect is lost. Concerning ongoing works, we are now investigating plasticity of resistance by observing the behavior of the same genotypes that are planted in different parts of Spain. For instance, here we can compare a plot in Madrid with a continental climate, with a plot in Valencia, with a coastal Mediterranean climate. After the first inoculation, we found a clear local effect of the location, with Valencia showing higher resistance than Madrid. And after the second inoculation in the same trees, we found that this effect is more or less lost, but we observe a quite high environment by genotype interaction. We are now studying 3 of these clones in terms of vessel anatomy, and we found that in Madrid, 2 genotypes increased their vessel area after the second inoculation, while the remaining genotype decreased their vessel area after the second inoculation. And interestingly, this was the most resistant genotype in Madrid, but not in Valencia. The behavior in Valencia was different, and for instance, the genotype here in blue decreased its vessel area after inoculation, and it was the more resistant genotype in Valencia, but not in Madrid. Other traits of interest are the area occupied by vessels, which was larger in Madrid than in Valencia, probably favoring pathogen dispersal, and the percentage of occluded vessels, which was higher in Valencia, probably favoring pathogen compartmentalization. Concerning the future, new resistant cultivars will probably be deployed in the next years. These cultivars come from crosses between these resistant cultivars. We have 2 plots. The first one was already inoculated with the pathogens. Here we can see the results. Quite promising, because many of these families shown symptoms below 30% of wilting. The second plot we inoculated the next year. We have selected or pre-selected 75 seedlings within these families, showing 0 to 5% of wilting symptoms, which is very good in terms of resistance. In spite, they show the typical browning of the growth ring, which indicates that the pathogen was present there. So we suspect that they are very resistant. So we have now to replicate it and to inoculate with enough replication. And to conclude my presentation, I just want to present the future works that are planned for the next year. We have around 70 new surviving genotypes, which are quite big, were detected in the field, pre-selected for pre-screening of resistance. These trees were located across Spain, from Asturias, north of Spain, to the south of Spain, Jaén, Andalucía, Málaga, Andalucía. We are also working in the construction of a reference genome of Ulmus minor. We are also performing transcriptomic analysis of resistant and susceptible Ulmus minor genotypes when inoculated with a pathogen. We want to study the interaction between M genotype and Ophiostoma novoulmi isolate, which is not very clear by now. And we also work to incorporate beneficial endophytes to plant production to observe if some endophytes improve the growth and the resistance of the elms in the long term. And that's all. Of course, many people in our group is working on elms, on different aspects of elms, from population genetics to ecology or physiology. You know? In name of all of them, I want to thank you for your attention. That's all.
Thank you, Juan. We have time now for some questions. We will have another, we can extend your questions to Juan in the following session of questions, but we have now 10, 15 minutes.
Thank you for the very interesting. I don't think so. This is working. Okay. Thank you for the presentation. I'm just wondering, you know, this various programs in the United States and Europe have been going on for decades, right? And I'm just wondering if something like this if a disease like this, it happens all the time anyway, but let's just say a disease like this happens today, with the new knowledge and tools that we have, how long do you think it would take to get to the stage where you are now with all these very interesting selections? In other words, can we significantly shorten the period of time that goes from discovery of a new problem to actually implementable resistant varieties?
Of course, we were very lucky because we had the support from the Ministry of Environment for many years. This is something that was not happening in other programs in Europe, or because, of course, traditional breeding needs a lot of time with elms. Elms should be inoculated, I mentioned, in the 4th year, so it requires quite a long time. I think that with this, for instance, this early screening method, we hope to be more effective in selection, at least discarding susceptible individuals. So we can concentrate in less material, less experimental plots, and it's a way to be more effective, probably more fast, more rapid in selections. I also imagine that in the future, with genomic tools and genetic tools, we will be able to accelerate the selection process. But by now, this is the way. We made the more difficult thing, that is the first selection of resistant individuals. Now we have some trees with quite good level of resistance. By crossing them, I'm sure that we will have trees that are almost not immune, but very resistant maybe in the next 10 years. But we are talking in terms of decades.
So are the bottlenecks the screening phase to find the resistant ones? What are the bottlenecks?
The bottlenecks? Is the time to inoculate, because you grow an elm, this elm should be planted in the field in an experimental plot in the soil. Pots should be avoided. This takes 4 or 5 years to grow, and then you have to inoculate 2 times. This is one bottleneck, and the other bottleneck is plant propagation, because if you have seedlings from cross, you are only inoculating 1 replicate. So you have to propagate to obtain at least 10 replicates of each genotype, plant it again in an experimental plot, and this takes another 5, 6 years. So at the end, it's a very long process. Now with in vitro propagation, we are obtaining more replicates, much more easy. With this pre-screening method also, we hope to shorten, but to be sure, you should be inoculate in the 4th year. So this is the bottlenecks: plant propagation and inoculation.
Thank you.
Hello. Yeah. My name is Ben Bumna, and I have a question. Your in vitro tests, Do we have any data that you can repeat them? You say you do it with 5-6-year-old plants normally. So can you confirm results from the in vitro test with your normal tests?
You mean the 5 years old?
Yeah, you do a test in vitro, and then you do it with your normal test in the field from 5-6-year-old plants. Can you confirm it?
Yes. The idea is to propagate plants in vitro.
Yeah.
Then we have thousands, maybe, or hundreds of plants. We perform the in vitro test, and at least we discard the more susceptible. Then the resistant trees we plant in an experimental plot in the field and wait for a year.
But are they resistant? The plants that are resistant in the in vitro test, are they resistant in the field?
Yeah, we want to confirm.
So you haven't yet. You haven't yet confirmed.
No, we haven't yet.
Oh, okay.
We haven't.
That was a question. Okay.
Well, when we developed this method, we used genotypes that were already inoculated with the pathogen. So we know we used some resistant trees and some susceptible trees. Okay. So this is the way we know that the technique more or less works well. But we now are in process of implementing this technique with new material.
Hello. Hi, I'm Soumya from The Ohio State University. First of all, this is very detailed and very great piece of work. My question was, did you at any point think that maybe since you have a good enough idea of the genotypes you have for both your resistant and susceptible varieties, why not establish a seed orchard and try something in the lines of breeding without breeding and genomic selection without actually having to do all the crosses, by the conventional method? Because breeding without breeding in some other species have helped, significantly reduce time, especially research related to resistance breeding. So my question was, have you considered that?
You mean to allow them to cross freely? We think this will happen, of course, in the plantation, in the natural plantation, and we will observe the behavior of these new generations in the field. But in the breeding program, we are not considering this option. But can be, of course, we trust that this will be the way that the nature will evolve in our plantations. And of course, new genotypes will arise. Some of them will be very resistant, some of them will be susceptible. But it's a good, interesting comment, so I will take it into consideration.
Of course. Thank you.
Just out of interest, my name's Jeff Williams. Yeah, sorry. I'm Jeff Williams. Work for the Forest Service in the U.S. I'm just interested, out of curiosity, over all of these years of working with elm, if there's just been any sort of other observations of other traits that may have not been what you were specifically looking for, that you've noticed were also associated in terms of, for example, we do a lot of selection for growth. Have you seen any relationships between growth and resistance, things like that, or other traits like seed set? Just out of curiosity, just...
Yeah. Well, the program is focused on resistance, but of course we measure also growth. And yes, we observe quite, not always, if the trees grows very well, there are no relation between age and resistance, but for instance here in Valencia, trees in Valencia I don't know if I can go. Here, trees in Valencia grow less than trees in Madrid. So this is one of mainly the causes of the resistance, because we suspect that trees that grow less are probably more stressed for for the environment, are more resistant to the Dachshund disease, because they form maybe smaller vessels, shorter vessels, and this impact the resistance level. I don't know if I answered your question.
Exactly half the thing I was interested in.
Any more questions? I have two. One, the first is that I am a little bit scared about the impact. The selective pressure that this disease caused on the population, because we're just talking about one, two, few genotypes in a whole population. The other was no resistant, and they was completely susceptible, and they disappeared. All that genetic information is over. So now you are doing this super nice job on these few genotypes, but maybe the question is: are you checking the cross resistance to another stressors, another pathogens, another pests in this material already right now?
Yeah. Well, first of all, susceptible material has not disappeared, because, as I told in the at the beginning, elms grow very well from as a recruitment trees from the root system, so we now observe a lot of elms. So elms are not in risk of extinction, so we have a lot of material that can be used for breeding. But concerning the cross resistance, we haven't seen. We are very focused on Dachshund disease, which is very hard. But of course it's important. We found that some of these trees that are planted in the field are infested by scolitids, quite heavy, and this is a problem, of course. And also other pests can be threatened, but this is a process that we hope to observe with these pilot plantations in the field, and we will know with experience. But I mean, the process is so we need to propagate so many plants, and that by now we are very focused on the resistance to the pathogen. But of course, it's very important what you suggest, and this we hope to evaluate this in the field.
Okay, thanks. And the other one is related to, you say several times that resistance looks like it came from the mother trees, no? You say resistance came from the mother trees, not for the side. So in the next days we are going to talk about this, maybe some epigenetics, some priming, some other concepts that can be underneath this region. Do you think that epigenetic marks or epigenetic mechanisms are relevant for the expression of these resistance phenotypes? I mean, if the resistance came from the mothers, maybe there is a transmission, transgenerational transmission of epigenetic signals or epigenetic marks.
I don't have an idea what are behind this process. We observe that it seems to work in this way, that resistance seems to be more related to the mother. Probably there are some traits, I don't know if vessel size or early response to the pathogens that are transmitted from the mother, but we have not investigated this, but will be, of course, very, very interesting.
Yeah, we have, there is evidences strong evidence that it's happening in annual plans, and maybe we can check this, start to checking these things in forestries too.
Yeah, that will be.
Thank you,
Great.
Juanan. So we are a little bit late on the program, but we will try, if it's okay for you, the coffee breaks are going to be mandatory at the time, you know. So anyway, we have free coffee line all the time. It's open for you. Just go have yourself, and and just I wanted to start with the first talk, and now I have to say thank you very much to all you. We know that was extremely hard to came here. I mean, a lot of things, the COVID, traveling regulations for everybody, budget constraints too. So thank you very much for your interest and for supporting this meeting. Second is thank you very much for our supporters and our sponsors. Can you? Our supporters and sponsors, supporters IUFRO, Misión Biológica de Galicia, our institution, and as well CSIC, which is our mother institution. And supporters, Deputación de Pontevedra, Concello de Pontevedra, the city of Pontevedra, helped us a lot. And mainly, and the main support is from Liceo Casino de Pontevedra, that they supported us since the very beginning. They give us this useful building, this saloon, all the facilities here, and I am I have to thank them very much, this support. And as well, the financial support from Xunta de Galicia, which is the government of Galicia, that with this Xacobeo 21-22 is supporting us, a very important part of the budget of the meeting. Xacobeo 21-22 is the pilgrim way, to Santiago de Compostela, happening every 9 years. There is a kind of a holy year, and for reaching Compostela, Santiago de Compostela by walk is a very well-known route coming from France, from Netherlands, from Italy, and as well from UK. So thank you very much for you and to our supporters. We can start with the following talk. And please don't forget to introduce yourself in your first question, okay? So it's time for Rómulo, think so? This is the first session, but try please to charge your presentations, posters, and talks in the session before your talk is. We have this screen over here, so the the speaker is you don't need to go back and to see you can see your presentation here, okay?
can I start?
As in the program, is I will suggest 13 minutes for uh, for uh, for being within the 15 minutes for each talk, taking account the time for going up and down, and so it's okay.
Okay.
I will show you. I will signal you 2 minutes, 1 minute. Okay.
Okay, fair enough. All right. Thank you guys for having me here. It's a pleasure. As you can see, my title is pretty generic, and that's on purpose, because I will not be speaking about my work specifically. I will be, it's not changing the slides. Okay. Nope. Maybe I should just tell you change slides, something like that. Yeah. Okay. Anyways, so my name is Romulus. I work for the Royal Botanic Gardens in Kew. As I said, I'm not going to be speaking about my work specifically. I will be covering a lot of stuff, so I'm going to have to rush to a lot of the stuff we're doing. You can change slides. So mostly, I'll be talking about the projects that are currently being covered by the Center for Forest Protection. This is a venue that was created very recently in 2021. You can change slides. And of course, it was created in order to protect the UK forests. The pilot year was 2021. Like my project is started with funding from this pilot year. There's a lot of work that I'm going to be showing that was started last year. But this year we also got funding. Those are just the projects that we're doing at Kew. There are more projects being done by the Forest Research, because the Center for Forest Protection involves both of these institutions. So we got a lot of fundings, a couple of millions, in order to just focus on trees. So it's a very good initiative from the British government. We're very proud of that. You can change slides. So this is just to show a few of the researchers involved. I will be focusing, of course, mostly on the work being done on my lab. There are way more people involved than these. But yeah, it's a big deal, and as I said, we are very proud of it. Can you change slides? So to start with, our lab is covering a lot of work with different trees. So I'll be covering each of these trees specifically. So to start out with oak, you can change. Louise has been working with a lot of sites across the UK. We have 2 different species of oak in the UK, Robbo and Petra. So she's been working with population structure analysis. We found out, this is a very simple result, but we found, for example, there's a lot of introgression going on between these 2 species. Even though they are not sister species, they're quite a bit apart in the phylogenetic tree. So we've mapped where the hybrids are located, which sites have more of those. We see that Robbers tends to introgress within Petrea more than the opposite, but both are going on. And Louise will be doing also some genomic environment analysis to see whether we can find some alleles related to climate change or any health-related alleles that we can find. She has started this work. It's still very preliminary. But our main focus, next slide, is acute oak decline, which is more interesting to you in this Congress. Acute oak decline is wiping a lot of trees in the UK. They can kill the trees pretty much within 4 years. We don't know exactly the causes. We know there are some bacteria involved. We know there is a beetle involved, but we don't know if there is any genetic basis to acute oak decline, for example. So you can change slide. So Louise, for example, has been working with metagenomics because we know there are at least 3 bacteria that are involved with this, are associated with the disease. But she wanted to know whether these bacteria are actually invasive and they're just killing the tree, or they're part of the microbiome of the tree and they're just exploiting weak trees. So she's been doing some work on that front. Can you change slide? And she found out, and this paper has been published already in Forest last year, that the bacteria that have been associated with acute oak decline, they are all over the place. So even in sites where we do not have acute oak decline, those same bacteria are present in the trees. So that suggests that something else is making these trees weak, and then the bacteria can cause problems for those weakened trees, for example, necrosis of the tissue. The same with the beetle, we think. The beetle do some gallery holes inside the tree, and if the tree is weakened, she cannot deal with the damage. I'm working with other people on GWAS, so the idea here is trying to find some genetic base and heritability for genes associated with acute oak decline. So these are the sites we're currently using, around 23 around the UK. We sampled almost more than 1,000 trees. We have 400 more to be sequenced. You can change slide. I've done some preliminary GWAS. I don't have those 400 trees included. I didn't find anything significant so far because I didn't have power, but we found some promising sites with a lot of LD going on that may be involved. For example, we know sites with acute oak decline have a lot of aluminium going on. There's some association there we know from other works, and I found an association with that. So there's some interesting, promising results. I'm going to be presenting a poster as well on that, so I can expand when it comes. So, yeah, for oak, pretty much we know there's a hybridization going on between the two species. Decline seems to be widespread, the bacteria, so it's not that, they're just infecting the trees. They are part of the microbiome. It's most likely these trees are just getting weakened by something else. And we have some preliminary evidence that there is some genetic basis for resistance to acute oak decline. You can change line. Now, on ash trees, which is something our lab has been focusing a lot. Richard Buggs is well known for his ash work, pretty much. And there's a lot of ash dieback in UK going on. They're caused by a fungus. You can change slides. And in 2019, that was before the Center for Forest Protection, we've done some work on resistance, and we found some alleles are associated with ash dieback, but you can change slide. But then Carrie wanted to explore there a little bit more and see, well, can we see whether natural selection is going on? If I go back to one of those sites we know are affected by ash dieback, are the juveniles that we find there more resistant than their parents? In other words, there's some change in allele frequency going on there, so that was her main thing. So she scored the tree health of the juveniles and the parents. She screened a lot of these trees, many millions of SNPs, 600 trees, 10 coverage in Martin Park Woods, which was a park we knew was affected by this disease. You can change slides. Again, this is unpublished results still going on. We have a preprint for that. I can give to any of you if you want. But she's finding first that the genomic breeding values, the breeding values are estimated through the genome of the juveniles are actually better than the ones from the parent trees. And also, if you look at the genomic values, they are a better predictor of health than if you just look at the canopy density, for example, because that's what pretty much we use to identify whether the tree is sick or not. You just look how much they've lost, how much dieback they have. But if you look at the genes, if you look at the genome, it seems that they are a better predictor of the health of the juvenile. If you know the genome of the parents, you can change that. This is also very interesting. To answer her own question, she has been finding some evidence for natural selection going on. There has been a bit of shift in allele frequency over time. This is just the effect size plotted against the change in allele frequency from the parents to the juveniles. And as I said before, the juveniles, they have a higher breeding value than we would have predicted had selection not going on. So it seems that this is very slight, the effect is weak, but we have suggestions that the juveniles are getting resistant to ash dieback, which is very promising, and it's a good build-up work from what we were doing before. We're also looking at structural variants. You can change slides. Daniel has been looking at structural variants is, for example, inversions, deletions, there are larger changes in the genomes. Most of these studies are dealing with SNPs, which is just one nucleotide change in a genome, but you lost a lot of variation and a lot of potential sites that could be associated with disease. So you can change slide. So he is looking at that. He will try and build a pan genome, which is a genome using a lot of different individuals in order to cover all the variants within those sites. So this is going on right now. They're starting to sequence. You need very high quality DNA for that. You can change slides. But he did a preliminary test with the data we already had from previous publications, looking at structural variants to see compared to the previous work with just the SNPs. And he found sites with the structural variants that were not identified before. So there's promising data there. You can change slide. So yeah, we know that there is some genetic base for tolerance to ash dieback. There is natural selection going on, and we can inform breeding programs with this information. Right? You can go. Finally, birch, we're working that as well. Mike has been collecting a lot of samples, like thousands. We have seeds already in the Millennium Seed Bank in the UK. He was planting this tree in Kew, and the idea, you can change slide, and the idea from all those sites is to do some genome environment associations. So you sequence all those guys. They're in different parts of the UK. They have different conditions of the environment there. So you want to see whether the genome, there are alleles, variants that are associated with specificities of the environment there. So that's the species that he's looking at. You can see it's quite a lot with sequencing at up to 50 coverage. So it's a lot of DNA, and we need a lot of storage for that. So we're grateful that we got this funding from the Center for Forest Protection, DEFRA. So we also, because we have historically a lot of provenance trials in the UK with birch, we can also compare what we are finding with the genome association analysis with what we already have for the provenance trials. So you have a lot of these sites. So you have European trees, but mostly UK trees. So they're planted. You can see whether each provenance, how they fare in different environments. So this has been going on for a while. And Mike, you can change the slide. Mike is also going to the sites that the provenance trials. He's collecting, sorry, samples from all those trees. And the idea here is just to cross-validate, right? He wants to see whether the trees are doing well on the provenance trials. Are they also the trees that he's finding in the GEAs that are, with some genes, variants associated with, let's say, water use efficiency, anything related to climate change and dieback. So, yeah, and this project is still going on. As I said, this project, most of them started end of last year, some of them beginning of this year. So he's already preparing these samples for sequencing, and he has collected like hundreds and hundreds. So, yeah. Change slides. So there's a legacy of work in the UK with birch that help us to cross-validate our results of genomics. So it's a very, very interesting interchange there. A lot of promising things to come. Can you change that? Yeah, and the overall conclusions, there's a lot of threats in the UK going on. Climate change, you have ash dieback, you have AOD. We're doing our best to kind of fend off those threats. And genomic research, that's the focus of our lab, will help to breed resistant trees, better understand disease and their consequences, and also choosing which trees to plant in which environment. Because if you know which provenances are better adapted to whatever environmental threat the place has, you can choose the correct individuals to plant in those environments. So that's pretty much what I had to say. There's a lot of people involved. I'd like to thank them. Why was I chosen among all this to speak about our lab? I don't know. Maybe perhaps because I've done my PhD, finished my PhD last year with sexual selection in birds, and they thought, well, maybe birds and trees are very similar. So that guy's probably the right man for the job. So here I am. Yeah, so it's been an interesting ride moving from birds to plant genetics, but I hope I did them justice. It was very simple, but it was a lot of work that I had to cover. So thank you very much.
Thank you get questions at the end of the session,
Yeah,
okay?
sure.
Please note your questions and elaborate the very best questions. Next speaker is Adrián from our team in Pontevedra.
Vale. No. Don't work. No. Okay. Good morning. My name is Adrián López. I am postdoctoral researcher vinculated to Católica Escola Superior de Biotecnología in Porto and the Museum Biológica de Galicia. I was talking about the evaluation of the fungicide treatments for the sustainable management of the pinaster trees infected with Bursophyllum cucillophilus. Now I, sorry for the things. Okay, I'm going to talk about a little bit the background of our teams. We are working in this with this pest from 2009. That in these years, with working with the four projects in relation with the Bursophyllum cucillophilus in different ways. One of these, the genetics, and the other in the using thermal treatments to eradication the Bursophyllum and goose pallets. And in this moment, in the present, we are working in a project. The name is Pointers. We go to finish this year. The Pointers is a project in relation with iteration in the trees and Bursophyllum, studying the alternative to fight with this pest. So in this year too, we have a new project. The name is BioVap, financed by the government of Galicia. This project is in relation with the endophytic bacteria used to fight to the pest relation with the Bursophyllum cucillophilus. The present is the Pointers. It's a multidisciplinary project vinculated with different foundations: the University of Coimbra, the University of Catholic of Porto, and Museum Biológica. This project has three main objectives: is to studying the nature of the functions of the proteins related to the nematode. The other part is studying the mechanisms of the resistance of the trees. In the end, it's in the developing sustainable strategies based in elicitors of fungicide practice for management on the pine wild disease. In the first of the studies to working with this pest, normally use the first control methods in relation with the pesticides and nematodes. These methods have a very adverse effect with the non-target organisms and the environment. For the reason, in this moment, all studies normally in the world are looking for the new method, control methods in relation with the biocontrol methods, because it's very good for the environment. For reason, we have two ways with this. Using the elicitors. The elicitors are biochemical products that to stimulate the defense of the plants. for two main ways. Using the salicylic pathway to produce the systemic resistance. If other way is use the jasmonic pathway. You produce the induced system resistance. The other way, we are studying here the fungicides too. Normally, these two fungicides, copper oxide and potassium phosphonate, is normally used in agronomy. We know that these fungicides have two properties, two nematicide properties, relation with the nematode. For this used in this new experiment. That in developing this project, we made three experiments. One of these is relation with the elicitors. In this experiment, they use one only kind of plant, the Pinus pinalter, is a susceptible plant to the nematode. Use three elicitors. Methyl jasmonate is a synthetic elicitor relation with the jasmonate, salicylic acid, and BTH. BTH is a synthetic elicitor relation with the salicylic. Okay? You use in this only pathogen, the Bursophyllus xylophilus. This experiment is actually published in Tree Physiology. Okay? The second experiment is talking about in this meeting is uses the fungicide, uses only one species too, Pinus pinalter, and two fungicides, copper oxide and potassium phosphonate. And use only the pathogen, the Bursophyllus xylophilus. This experiment, in this moment, it is writing the last paper to publish it. The third experiment is actually to doing the analyze the dates and finishing some analysis. Sorry, this is a relation with the studying the jasmonic pathway. For this study, you use three different species: Pinus pinalter, is susceptible, and Pinus pinia, and Pinus ptaeda. Pinus pinia and Pinus ptaeda are resistant to the nematode. For this, you use the methyl jasmonate, is the elicitor, and you use an inhibitor this pathway, the YACA. And use the only one pathogen is the Bursophyllus xylophilus. Now I'm talking about the second experiment, relation with the fungicides assay. Our hypothesis here is that if spraying the plants in greenhouse with the solutions, the copper oxide or potassium phosphonate improves the pinalter resistance against the nematode. For the objective is to demonstrate our hypothesis, we want to study the efficiency of the fungicides, studying the evolution of this disease, studying the defensive capabilities of the trees through various processes for damage, and studying too the modification on the endophytic bacteria. For this, to conquer these objectives, we measure different variables. One of these is number of nematodes and foliar symptoms. The other is concentration of photosynthetic pigments, phenolic compounds, melanoidin day, and bacterial populations. He has discussion about the methods of our experiment. It's a lot of information in a small space, but I'll explain very, very quickly. We use 117 trees separated in 18 non-infected, non-treated control plants, 33 infected non-treated control plants, and 66 infected with treated with copper oxide, and the other part with potassium phosphonate. In the conquer, the conquer of the right conquer of this slide, we have to the products of the reproduction of the nematodes in lab. It's important because you need a lot of samples, a lot of inoculum to use in the plants. For the reason you use applicates with Botrytis cinerea, and this applicates you reproduce the nematode. When you have a lot of nematode, you can start the inoculation assay. You put the samples of nematode in the burning funnel, that in 2 to 4 hours, and then you can inoculate the plants. And you have the inoculated, treated, non-inoculated. For these plants, you use to get the samples, the leaves and stems, and to deep frozen and use for the biochemical analysis or for use the content of the nematodes. We have some results of this assay. We have in the first the number of nematodes. You can compare in this graphic this in the top the number of the treatments, in the bottom at the time. You can compare that is significant all treatments in all times. You can see the compared difference in the last time, 35 days after inoculation. We have more numbers of the nematodes in 2 treatments in comparison with the no treatment. In the other graphic, we have other foliar symptoms. In the top, the stage of damage from 0 is healthy and 4 is death. You can comprobate that in the 4 days after inoculation, any symptoms comprobate in the plants. After the 21 days after inoculation, start the symptoms. Normally, you can see in the first column is no treated and non-infected, and the rest are infected plants. Okay? The first column is the infected with treatment, and the 2 is with treatment con 2 fungicides. You can provide in the last day, you have difference between there. Normally, in the infected plants without treatment, we have a lot of symptoms in comparison with the other plants. You can contrast the color of the columns with the stage damage. Could provide the difference between these. Here we have an analysis of the biochemical compounds. In the top of the slide, you have the anthocyanins and flavonoids. Normally, there are compound relation with the resistance of the plants with stress, biotic and biotics. We have comprobated with that is significant use the treatments and the time too. In the bottom in the slide, you have the phenols and MDA. Normally, these biochemicals are in relation with the stress, but the production of the plant, not the defense. Normally, where you hide the stress, hide this biochemical. In this case, we have to significant between treatments and time here. The other part, we are studying to the difference in the endophytic materials between different treatments. You can see in the bottom of the slide the difference between the plants non-infected, not treated, when plants infected and not treated too. You have different species there. We can see here too difference between the plants infected, not treated, with infected with treatments. You can see the difference between. Normally, the only species common in your own cases is the Pseudomonas florens in this case. For the answer conclusions about our study, we can conclude 3 main conclusions. The application of the fungicides reduce the damage caused by the nematode, reducing the progressing of the disease, but without actually stopping it. This is important because we just reduce the infection, but don't stop it totally. We have this experiments in lab. We use a lot of number of nematodes in comparison within nature. For the reason is this thing is important. Okay. The second main conclusion is there are biochemical differences both between treatment and untreated plants, which indicates the different effectiveness of each of the fungicides in activating the defense against the wild pine nematode, in comparison with the last graphics. And for the third main conclusion is that endophytic bacteria are altered by the both nematode infection and the application of fungicides. For finish my meeting, I'm talking about the relative future, because this this project is starting this year in May. It's my first project by PE. It's complicated, but it's good. Speaking about our future project, our hypothesis is that after the studying to all things in my postdoctoral thing in Católica, I'm talking about with my boss and my mind, and I'm trying to describe a new project in relation with the information in the past. For the reason, and our hypothesis for our new project is, I think, the variety in endophytic bacterial communities between pine species explains the difference of each species susceptible to Phyllophilus. For this reason, we have the identification of the antagonist bacteria engaged on the nematode in these communities. This identified material can to improve the pinaster resistance against this pest. For the reason, in the project, we are studying the bacterial in 3 different species. The pinaster is natural pine from here and susceptible. Pinia is natural from Spain, but no from Galicia. It's resistant. And Pinostera is a species original of the nematode. It's a combine between the nematode in America. Studying this to conquer this objective, we have to study the 3 different experiments in relation with this project. One of these, endophytic bacteria on the infected and the non-infected pins with nematode. Second experiment, using this bacteria identified analysis in vitro antagonism between the identification bacteria and the nematode. In the last experiment is studying the resistance of Pinus spinaster cultivated with the antagonist bacteria of nematode. Okay. Okay. Thank you for your attention. Here have the title of the paper to publish in Tree-Physiology, the relation with the elicitators. You have my email for any questions. Thanks for invitation here.
We decided to, Adrian, you can stay here. Okay. And where is Romulo? I think it's time for questions for these 2 speakers, and then we can go to the coffee break. We have just a few minutes for. Can you move the back?
Should have that be my question?
Don't go anywhere. So thank you very much for your presentation. My name is David. I'm representing basically British Columbia, Canada, Ministry of Forest. So my question is, did you consider health-related issues with copper oxide when you're using as a fungicide? Because how you came to this conclusion to use fungicide, this type of fungicide against that? Because I'm more for your objective tree, like using biocontrol agents against this disease than using fungicides like copper oxide, because it's harmful for ecosystem, animals and plants. I read some articles about it. The thing is, we were using this, some of our companies were using Bravo chlorine-based. And at the end, it didn't control Swiss needle cast on Douglas fir. It harmed more than helped our system. So my question is how you came up with this conclusion to use that fungicide for your research. Thank you.
Sorry, don't work. You listen to me or yeah? Or no? No work? Hola? No? Yeah? Oh, sorry. Yeah. Oh, sorry. They're using this. The question is, you're using this in the countryside now or no? Relay? I understand you. Sorry. I think. Sorry, now I listen you.
No, I mean that. How did you reach to this conclusion to use fungicides against that? And did you consider health-related issues with this type of fungicides?
Yes, human health. Okay.
He means human health,
Okay.
environmental health.
Okay. I think it's a preliminary study in relation with this, but to apply the countryside is in this moment a bit complicated because normally the forest don't use the biochemicals. For the reason I think it's better to use to the greenhouse, to seedlings, to take with this in the first or second year to elicitate or you can reduce the infection with the plant. But in the countryside, we need more studies with this. But for this experiment, you need more money to work with this kind of this. Normally, to just our teams to working in the greenhouses to elaborate the new protocols you want to prove in the countryside. But in this moment, in the preliminary, to use to this. Sorry.
Hi, good morning. Marcin Nowicki from University of Tennessee. Romulo, in your study, you had shown nice variability in oaks. From the species that we see and that we study in Tennessee, we see quite a variation in the genome sizes in the oaks. So my question is whether you observe that for your species. And second, following that, is how do you account for that in your research for GWAS or other analysis?
So as I said earlier, I'm only going to accept questions about birds. Plants, no. Sorry, so I didn't get the last part of your question. The genome size of the one I'm working with Quercus robo only. I'm not studying Petria. So Quercus robo has roughly 750 gigabytes of a genome. We have an assembled genome. Actually, there's one that is coming out now from the Darwin Tree project, which is going to be probably better, because the one I'm using, there's some scaffolds that haven't been assembled. So yeah, I'm only working with one species, and I don't think in GWAS, as far as I'm aware, taking into account the size, because I'm not comparing different species, then I don't think it's relevant.
Right. The question is, I guess, whether this genomic variability or genomic changes could affect your effects, whatever you're studying using GWAS, right? The changes that we see for our species are substantial, so it's not like we're talking, couple thousand base pairs. We're talking actually substantial 10% or up to that using flow cytometry within a species. So there is a lot of variation for the white oak, for instance.
Yeah, that's why ideally you would like to have like 20,000 individuals, right? But we have to do what we have, and we're trying. As I said, we're going to add 400 individuals to this analysis, and precisely because of the difficulty, and so there are so many SNPs I've sequenced, I've called like more than 1,000 million SNPs, whatever. So what we're probably going to be doing on top of the GWAS is trying some genomic prediction as well. So we're going to do other analysis in order to see whether this variation we're picking is actually something causal or is just, just chance. But as you can tell from that slide, the power that I have now is just not sufficient. So I cannot say confidently that those SNPs are actually doing anything. But it's promising because some of the stuff we found have been associated with AOD completely independently by people just studying the ecology of these plants. So it's promising. But yeah, you're right. There's a lot of variation, and you have to be careful with GWAS not making too strong claims. So we're going to do other analysis to back up whatever we find.
I'm going to take advantage of the microphone prerogative. My name is Enrico Bonello. I'm from Ohio State University. 2 quick questions for you, Romulo. I did publish a paper with Richard Buggs on European ash resistance to emerald ash borer. Do you know if any more work is going on in that arena? And second, have you guys thought about, or does a genome of a European beech exist, or are you thinking about doing that because beech leaf disease is going to come to Europe?
I think the work we're doing with ash is the 2 works that I've presented here. So we're looking at natural selection, and we are trying to do this pan-genome precisely because getting structural variations from a linear genome is not easy. So that's the 2 ones that I am aware of that we are doing it. If Richard is doing something on the side with other people, I'm unaware of. Your second question was? That's a good question. I think from what I spoke to Mike, I don't think all the birds that he's using have been assembled. Beech, yeah. Fagus. Yeah, I don't know. I have to look it up. But yeah, I think he's not. I don't think there is reference genomes for all the plants that they're studying. So they're having to use whatever species that has been assembled. And we don't have funding for assembly. So we're not going to be doing any assembly of genome, only the pan genome for ash. $150. Yeah. I'm not hearing any bird questions at all. But anyway, so sorry. Your first question was whether I'm looking at LD. Yeah. The reason I went quickly because I'm going to present DOD work in a few minutes, so I'm going to be repeating a lot of stuff. But I've looked at LD. LD decay in oak is pretty fast, actually. So after 200,000 bear pays, you don't find any LD. So you have to, yeah, it's part of the population structure analysis. We do all those LD studies. The details about each one's projects, I don't know, but I'm pretty sure that's just so standard that they will be looking at LD. And we know, I just came from a workshop in Paris with GWAS, and there's some stuff that have been neglected in the literature. So for example, LD will vary depending on the allele frequency, and your models have to account for that. And a lot of GWAS do not account for that. They assume the LD is constant, whatever. So yeah, I attended a workshop, and I'm planning on using the information that I learned there in my project. But I do have LD plots in my poster. You can look it up. Well, there's a blue tit that keeps singing on close to my bedroom, and he wakes me up at 4 o'clock in the morning from time to time. So we have a love and hate relationship, so I would say blue tits for now, yeah.
I think it's time for coffee break. We try to start in time after the break. Okay. It's upstairs, the first row. this Jeff is around, now going out from the Camerino.
One. Bueno, gracias. ¿Listo? Okay. Everyone, my name is Jeff Williams. If we haven't met yet, I currently work with the USDA Forest Service in International Programs Office, which is a part of the Forest Service that does a lot of different types of forestry work all over the world. But I'm going to be talking a little bit about some of what I was doing over the last 6 months in Argentina. So I actually came directly to the Portugal meeting from Argentina, where I was for the last 6 months. This is just a picture of ponderosa pine, same age, probably the same seed lot. I'm pretty sure this is aphid damage, but you can see that, but I don't know for sure, but you can see that there's pretty much none of these signs or symptoms on the tree on the right, but this is just a cool picture. So the title of my talk is Closing the Gap Between Forest Pest Emergence and Global Change, a case study of international exchange of expert local knowledge. And I'm going to be talking about invasive species. Here I'm talking specifically about insects or pathogens for which hosts show little or no resistance that are typically introduced through global trade, and they cause unsustainable losses, whether they're economical or ecological. Some examples that we all know about: emerald ash borer, pine wilt disease, Cyrex wood wasp, which is really important pest of pines planted in the southern hemisphere where they're exotic, and of course this aphid Cinara cupressi, which causes a lot of mortality in native Austrocedrus chilensis forest in the national park near Esquela, where I was based. And of course, one of the major issues here is it's hard to get ahead of these things because there's always a lag. There's often a lag in detection and response. So we're trying to think of proactive ways to protect forests. So we're all familiar with the disease triangle. I added insects onto one side, and I just wanted to talk about how these types of associations have changed in recent years. So this is you can make a much more comprehensive list of things, but here I just have life history and genetics of the agent, the host, its ecology and genetics, and then environmental impacts on pest performance or on the host's performance. And in order to have disease or an invasive species that really becomes invasive, all these things need to fit together in a certain way. So in most cases, they won't, right? So we're looking for the cases where they actually do. And historically, there are barriers and there are buffers that kind of limit this. So when I'm talking about barriers, I'm talking about dispersal barriers like oceans, and also climatic barriers that maybe limit pest damage in certain parts of a host range. And then in terms of buffers, these are processes that feed back on pest performance and on these outbreaks. So this could be natural enemies, it could be competition. Also in natural forests, the disturbance history caused by pests and other disturbances will influence forest structure and diversity, which will have feedbacks on, we see a lot of these problems in plantations where we have, lower diversity. And then of course there's evolution, there's genetic feedback. But in the Anthropocene, we have a lot of breakdowns in these barriers and buffers. So global trade obviously eliminates a lot of dispersal barriers. The introduction of new hosts to new environments, something that's going to continue. and then forest degradation, and of course climate change. And this leads to a lot of novel pest problems that we haven't seen before. And just as an example, the pinaceae is mostly native to the northern hemisphere. As you can see today, it's widespread in the southern hemisphere. And this introduction of hosts to new environments may facilitate adaptation and host range expansion of pests. And this is likely to continue as we move genetics or even species to new environments as parts of nature-based climate solutions or just afforestation and commercial forestry worldwide. And we can take advantage of this to study potential pests that have not yet made it to the native range of host species. And this is what we're referring to when we talk about ex patria sentinel trees, which I think is a really fancy word. But it basically is, it just means trees that are outside their native range that can be monitored for emergent pathogens. So for example, I have a picture of ponderosa pine in its native range, and then in Patagonia, where it's not native, or also in North America or in Europe, you may see Araucaria, which is native to the southern hemisphere. So by studying these interactions outside the native range of hosts, it facilitates the development of more proactive responses, whether that's selection for resistance or understanding biocontrol or whatever. And one of the keys to these types of international collaborations is to really talk to people about what they've observed in their careers, whether they're pathologists, forest managers, just to understand what are the real problems. And so, and then as part of this work, I also was asking, you know, have you seen any variation in the severity of disease in exotic trees? And I just have a picture of a couple of my collaborators in Argentina, Belén Pildain and Andrés de Arasti. I was based in Argentina. For the last 6 months, I was in Esquel, which is in the Chubut province of Patagonia, Patagonia is the entire southern part of Argentina and Chile. This is a typical landscape that you might see in the national park just outside of Esquel. And actually, my Fulbright project didn't have anything to do with exotic trees. I was actually working in native forest. Oh, those are exotic trees. We were working in Coihue and Lenga, which are two species of Nothofagus. I just wanted to tell you about this so that you know that, you know, my work there wasn't just self-serving for the United States, right? I mean, I was actually there to work in native forests, and that really facilitated the collaboration. And we were studying gap mortality phenomena that you might see. Oh, man. There was a picture that was supposed to come up there, but yeah, so I brought a mint ion with me. We did some meta-barcoding studies with soils and wood, and we were also studying a genus of ambrosia beetles that's very diverse in these forests. Some other trees that you might find are Ostrasagus tolensis, I mentioned before, and Fitzroya cupressоides. These are both in the family Cupressaceae, or Cupressaceae, however you want to say it. There's also a lot of other conifers and gymnosperms in this region, but there are no pines that are native there. However, the most abundant species of pines that are native there, and they're a very significant part of the landscape, are radiata or Monterey pine, lodgepole, and ponderosa pine. There's also quite a bit of Douglas fir that's been planted, especially in the last 40 years. And there's a wide diversity also of cedars or Cupressaceae everywhere, but mostly in urban areas. And before I get into this, I just wanted to talk a little bit about the history of afforestation in Patagonia. It's a little bit of a controversial topic. Many people don't like the pines there, with good reason. Early on, there was a lot of replacement of native forests to produce pines. More recently, the species that have been introduced do really well, so they're highly invasive, and there have been some pretty severe forest fires, probably as a direct result of the introduction of pines. But they're also important they were to be important for production of timber, but the industry is not really there these days. That said, prior to the 1930s, the major species that was planted for plantations was Monterey pine, had some issues with producing quality timber because of the European pine shoot tip moth, so they would lose their leaders and bifurcate. They didn't be able to get good wood that way. Actually, in the 30s, I learned that Theodore Roosevelt traveled to Bariloche and really advocated for the establishment of national parks. And this actually led to a lot of introduction of North American and European conifer germplasm, most of which initially was propagated on an island called Isla Victoria. And there's now exotic forests that are 100 years old, very diverse conifers there. And a lot of the initial genetics on the landscape came through that nursery. And a lot of the initial plantation level forestry was with lodgepole pine. But in the 80s, there were government programs to introduce more ponderosa pine and Douglas fir. There hasn't been a lot of management since then, but the younger plantations are mostly those two species. That said, lodgepole pine and ponderosa pine are very diverse, especially ponderosa pine. From consulting people and looking around, at one time in North America, we called any orange-barked pine tree a ponderosa pine, but there's actually a dozen species that can all hybridize, and even species outside of ponderosa pine. Some things we call ponderosa pine we don't even include in ponderosa pine. I'm aware of at least these 3 major subspecies, and then these other 2 species. You may be familiar with Pinus scopalorum. This is kind of like the inland ponderosa pine. But based on the one genetic study that I've found, which did not include any molecular work, it seems like the initial seed sources for the ponderosa they were working with in that study were from near Truckee, California, which is an area where there's some kind of sympatry between subspecies ponderosa and washuensis. So these are both West Coast subspecies, and most of the pine that I was seeing around Esquel is probably subspecies ponderosa. But like I said, consulting with people, seems like there's a lot of stuff. So I developed this key. Not going to talk about it. I didn't really get to test it out that much, but that was one of the ways that I was able to kind of tell that most of what I was seeing was subspecies ponderosa. So, like I said, I think I said all of this, but they do have a improvement program that was established with ponderosa pine. So there are seed orchards, especially in the region that I was working, that may present some opportunities. So I'm just going to shift gears and give some background on my Forest Service project. So we're really interested in knowing what's already known about pests and pathogens that affect host trees outside of their native range. And I've developed a system that takes advantage of a number of different databases, none of which actually focus directly on forest or necessarily on pests and pathogens that are invasive. And basically the way it works is you can put in a host species. You start by getting all of the host pest associations that are known, and you can add by annotating this database. And then it's totally reproducible, and then you can also download all from many different databases the distributions of those pests and figure out which ones are known within the native range and which ones are not. And then the most important step is then consulting with experts and literature to understand which of these are really posing a problem and which ones are just reports or maybe just not to be taken as seriously. And using this system in Argentina, there are a few species of fungi, bacteria, and other agents that are not known in North America. 5, 10, 8, and 4 for these 4 pine species respectively. But of those, I think the thing that they deal with the most as a management issue is Cyrex. And Cyrex noctilio is present in North America, but in Argentina, it can cause up to, it's currently still causing up to, in some cases, 80% mortality in stands of ponderosa pine. Some of this has to do with management. There's lots of other factors that could be involved in terms of whether that would be a problem in the US. But they have a long-term monitoring program that they have a lot of data on this. And if you look at the native range or the invaded range in the US, We really only have Cyrex noctilio in the Northeast, as far as we know. But lodgepole and ponderosa pine are really important components of western forests. So I think we need to be paying attention to this. And one of the ways we can get ahead of that is actually potentially by supporting development of resistance in these species outside of their native range where Cyrex wood wasp is a big problem. And that's all. I don't really have any results, but this is just, most of the work I was doing was meeting people, forming relationships, and I think this is really the most important step in doing something like this. So the take-homes are, it's helpful to think about host and pest distributions within the native and outside the native range, like together, and not just always restrict to one to the other. And the value of sentinels isn't restricted to things we don't have yet in the US because we're still able to observe these associations where they're happening, right? And they're not happening in the US. So that kind of has opened my mind a lot, seeing Cyrex as an opportunity. And I just wanted to emphasize that there may be synergistic opportunities for genetic improvement. So, I mean, by virtue of supporting research in native forests, I opened up the opportunity to also be consulting about, how can we also take proactive approaches to protecting our interests in the U.S. with the health of our forests as well at the same time. And I just want to thank a lot of people, but especially these institutions, INTA, which is the USDA, basically their version of the USDA in Argentina, CFAP, the institution where I was based there, which is part of their National Research Council, the Forest Service international programs, Fulbright, and then the University San Juan Bosco and Esquel, and a number of other folks. And thanks for your time. And that's all I have today. So, thanks.
Your questions at the end of the following session. So we are moving your question for the end of the morning. So, I think particularly these sentinel plantations and this approach can be one of the topics for thinking during these days. We should write or an opinion about this thing. Is that my personal opinion? I mean, so we are going to try with the poster sessions. Can you go up, all the people in this session with the posters? We can stay here and just present. Francisca is the first one? No. Karina, okay. Yeah. What was you? Okay.
Sorry. Can you help? No, here it is. Okay. This one?
Yes. All right. This is too tall for me. Sorry. So you're gonna hear my voice, but not see my face, probably. I'm teasing. I'm fine. It's a no. Okay. Yeah, I like this better. All right. So this is the poster. You can find it over there. I think it's the first one near the coffee, so you might go there often. It's the work of my student and lab coordinator, Colton Maneki. And I'm saying up front, this is actually a collaboration that he's doing with the University of Florida collaborators. So I'm very marginally involved with this project. So for every question, ask Tanya over there, who's more involved in the specific project. And the project originated after they realized there is a need for accurate prediction and speeding up the resistant breeding for slash pine, which is a very important commercial species in the south of US, especially in Florida. And so the objective of the project is to develop selective models using both chemical and genetic markers together, integrating them for a quicker and more accurate prediction of resistance. Now, this is a project that just started. They received the funding and they are just at the organizational part, so there's no result. It's more of a proposal type. And I'm going to go very quickly over the overview of what they're going to do. They already isolated many different isolates of Fusarium tercinatum, pitch canker, one of the major problems for slash pine. And they have many, many families of slash pine, and they're inoculating those in a greenhouse setting. And they're going to phenotype them for resistance based on the lesion length. But at the same time, they are using nanopore technology to sequence the genome of the fungi, all the families, and also NIR technology, which maybe Enrico will talk better later on, to get chemometric models for phenotyping resistance. And so by integrating all this data together, the genetics of the fungus, the genetics of the plant, and the chemistry of the plant together, they hope to get a selection model that are more efficient than the technique that they're using right now. And that's all I have. It's a poster, so it's over there. And ask Tanya for question. That's my take home.
Hi there, I'm Franziska Paas. I'm from the Thun Institute of Waldseewarsdorf. I work in the resistance department of our institute, and I would like to give you a short presentation of our project Res Esche. Res Esche is short for resistant ash, and the topic we were dealing with in this project is ash and ash dieback. I'm sure many of you have heard already about this disease, but I would like to give you some basic facts just to bring the matter back to mind. The ash is a very important tree species, especially for the ecosystem and for the timber industries, for instance, for the tools and for the sport equipment industry. The ash is endangered by the dieback-causing fungus Hymenoscyphus fraxineus, which probably originated from Asia and has spread through Europe since the early 1990s. In Germany, we first found and confirmed the fungus in 2002 in Mecklenburg-Western Pomerania, the very northeast federal state of Germany. And basically, this fungus is causing necrosis, which damages the crown very severely, so that the tree is getting weaker and weaker until they finally give up and die. What is interesting is that in those heavily infested areas, there are always trees which seems to be pretty healthy, and it is assumed that they have some sort of resistance within. And these trees are very valuable for breeding. And in our project, we focused on those healthy-looking trees. Basically, our goal was to create a seed orchard with those tolerant and tested genotypes. We conducted the project with our partner, the Forest Department of Mecklenburg-Western Pomerania, and we realized this project by searching for those healthy-looking trees. We created grafts. We tested those grafts on their resistance by 2 infection tests. And if those genotypes survived the infection test, we were selecting them for the seed orchard. We started the seed orchard in 2019 with the first couple of genotypes, and we were adding more and more genotypes up until 2021. And we now have 126 genotypes in our seed orchard, each represented by 5 to 15 grafts, and around 40% of them are female. And this project can be seen as kind of a small-scale pilot study for the now running Fraxgen project, which uses similar techniques but on a larger scale, and my colleague Ben Bubner will tell you more about it soon. So that's it from me.
Well, I'm starting. My name is Raquel Díaz. I work at the Forest Research Center of Lourizán here in Pontevedra. It depends on the Xunta de Galicia. I'm going to talk about the advances we have, the Galician breeding programs of Pinus pinaster and Pinus radiata against or to control pine wilt disease. Yeah, it's quite hard to see it right here. Well, the nematode is the Bursaphilencus xylophilus, and it is monocamus insect to go from one tree to another, and a host, a susceptible host, which is a kind of a conifer and mainly pinus, This photo, you can see the, some trees dying because of the disease. I'm going to do this way. And here you can see the situation right now in Europe. Portugal is the demarcated area, and in Spain we have three main spots. One of them is here in the south of Pontevedra. Okay, we have done several work on this subject, but I'm going to present here three parts of it. One of them was to study the variants with different strains, two from Japan, two from Spain, two from Portugal, and one from the USA. And we saw that there were significant differences among the strains, and the Galician one was one of the most violent ones. So we used this strain with to test for resistant genotypes in our breeding programs. The work we did with Pinus pinaster is shown here. We evaluated 91 families from our breeding population, and we found significant differences among families with a heritability of 0.25. So this searching for resistant genotypes can be a good control method for managing the disease. The best or the more tolerant families had 78% of survival, which was 25% over the average. So we included them in the Spanish Forest Catalog of Reproduction Material because we need to do this in order to sell these plants in the nurseries. About Pinus radiata, we can say that we have done also several assays, and we have tested 55 families, and we've had the same result. There are significant differences among families with a heritability of 0.43. So tolerance to this nematode can be included as a control measure to fight against this disease. The best the most resistant families, the best 7 ones, had a survival over 65%, which was 35 over the average. Well, we are working right now in several points, which one is the phenotypic genotypic study to search for candidate resistant genes. We are doing control process, and also we are doing clonal resistant assays to search for most resistant genotypes. And we want to continue searching for reliable, not destructive fast and evaluation methods, and to continue searching for nematophagous fungus to help controlling the pineweed nematode. We are going to talk about this on Thursday. And that's it. Thank you very much.
Okay, so we are going to start now the following session. Maybe Malin can help us, some moderator. No, I think we are going to follow with the next three talks, no? Yeah. Yeah. Now is the time for Heather Dunn. It's no?
Yeah, the microphone.
Oh, I can hear it, yeah.
You know,
Yep.
we are quite okay in the program because there is some colleagues coming from South Korea that miss the plane, so they let us know that they are not able to give the presentation this morning. So we have some slots. We are more or less okay.
Now it's on. It's definitely on. Okay, so now we'll kick off the next session with Heather Dunn talking about Phytophthora rumorum on Japanese larch. Then we will do one more talk and do the questions on these two talks before lunch, if we're lucky. If we're not lucky in time, then we will do the questions after lunch, because lunch is apparently 1 p.m. sharp. So it's up to us. So please, Heather.
Hi, so I'm Heather Dunn and I work for Forest Research in Edinburgh, and we're going to present some of our work that's recently been published, which is a combination of some of my PhD work from the University of Oxford and also work we're doing at Forest Research in the pathology lab with Sarah Green. So I'm going to present on exploring variation and susceptibility to Phytophthora rumorum in Japanese larch. So we'll start, oops, whoa, that was a speed talk. How do I get that back? There we go. So we're going to start with some background. Phytophthora rumorum is an oomycete pathogen that's most well known for causing death in oaks in America, but in the UK it affects larch instead. And as you can see from the picture, it can cause bleeding lesions, which then girdle the trunk and kill the entire tree. So it can be moved by water and wind. So we have the interesting phenomenon where it can infect from the base, girdle a tree and kill the entire tree, or it can infect aerially and work its way down. So we can die from the bottom up and the top down. Larch is also unusual because it's a sporulating host. So usually the hosts that get killed by a pathogen don't produce a lot of sporulation, but larch is an incredibly effective sporulating host, and there's a high inoculum load in the canopy that then leads to epidemic spread. There we are. So it was first seen in the UK in 2009, and then over the following years it moved up the west coast along the damp corridor of high rainfall that we have there, and in 2013 it ended up in the Galloway Forest. The Galloway Forest is also unusual because there's a new lineage, the EU2 lineage, which is even more virulent on larch than the EU1 lineage that was found in southwest England. So throughout this talk I'm going to be talking about the EU2 lineage. Are we moving? Am I pointing at you instead? There we are. So the importance of larch. Larch is our third most important conifer tree, after Sitka spruce and Scots pine, accounting for 126 hectares of plantations before it started dying of rumorum. Since rumorum's become a problem, we've lost about 21,000 hectares, either through death from the pathogen or preventative felling. So the current management policy is to fell all trees that are infected and all trees within a 200-metre barrier, which leads to a lot of felling. Larch is important because it produces good quality timber in cold, wet upland sites, which we have a lot of in Scotland. And the only other alternative species that we have for that is Sitka spruce. So as you can see, our system in the UK predominantly relies on Sitka spruce. It accounts for 51% of all conifers, and in Scotland, Sitka spruce accounts for 45% of all trees. So if we lose larch, we instead have to plant more Sitka spruce, and that increases the fragility of a system that already predominantly relies on a single species. So there's a lot of interest from industry in maintaining larch as a commercial conifer species, because otherwise they'd have to plant more Sitka spruce. So our research site is the Galloway Forest. So the Galloway Forest has a large area of larch, and when the EU2 outbreak occurred, there was so much larch that it couldn't be felled as normal. So you can see here at the top in 2012, all the bright green trees are larch and the dark green trees are Douglas fir. And in 2013, you can see the mortality throughout the site, with the Douglas fir left alive, but most of the larch is dead. As there was so much larch, it exceeded the capacity of the industry to fell it, so instead a management zone was created where the trees would remain standing until capacity was available to fell them, which allowed us to retain the stands for research instead of them all being felled as had been carried out throughout the rest of the UK. So we went looking for survivor trees. So in areas of high mortality, so you can see here we have almost complete mortality of entire hillsides, we would find occasional survivor trees which had good healthy crowns. And it was thought that these trees might have some level of resistance, or it could just be that they had survived by chance. So in 2015 we managed to identify 20 trees, and in 2018 we identified another 50 trees. And when we found these trees we had them felled and collected soil material for grafting onto rootstocks. So we then carried out inoculation of clonal ramets. So you can see here we've got them grafted onto rootstocks in a polytunnel. And we have, from our 20 trees in 2015, we have two groups. So one is our resistant group, which are survivor trees that when we had them felled, they had absolutely no signs of infection in the crown. And we also had a susceptible group, which was when they were felled, there were some signs of infection in the crown, but they were still surviving. And we carried out inoculations with a zoospore sporangia suspension mix, which we thought better replicated the natural infection pathway in the wild. So we inoculated onto a little wound, as you can see in the middle here, on some excised shoots, and incubated them for 3 weeks in an incubator. And you can see here on the left, we have the red arrows showing the inoculation point and the black arrows showing the extent of the lesion development. So we had good lesion development within the excised material. However, before we looked for lesions, we looked at the exterior of each shoot. You can see the controls here are bright green and have good needle retention. However, there are differences within the clones. So you can see in the centre, these are individuals that develop short lesions, and although they are discoloured, there are green needles and there's good green needle retention. Whereas if you compare to the far side, these ones developed the longest lesions, and you can see there were a lot of needles missing, and there was also a lot of discoloration and general unhappiness. So if we then look at the differences between clones, so along the bottom are the different clones. So SUS is our susceptible group and RES is our resistant group. And you can see that we have a continuum in lesion development from very long clonal development to short clone development. And although there is a continuum, there are significant differences between the shortest and longest lesion development clones. However, there is no significant difference between our two groups. So our resistant group and our susceptible group are not significantly different. But as you can see here with the resistant group highlighted in yellow, they tend to be more towards the short lesion development side of the spectrum. So although there's no significant difference, we would like to propose that using trees that are survivor trees and also show absolutely no signs of infection provides a good resource for further work. As this is kind of the first investigation that we haven't done any further genetics on this, but this is providing the basis for our further work. And all of this has been published in Forest Pathology. From then, we moved on to our second group of clones, which we inoculated just this past summer. And you can see highlighted in yellow, there's S2 and S3, which are one of the long and short lesion clones from the previous group that we've included to allow us to compare between the two groups. So again, we have a continuum of lesion development with significant differences between the shortest and longest lesion development clones. However, we have longer lesions than we had last time, but we haven't found any trees that are better than the best ones we found in our group of 20. But again, this gives us more material for further work. We also know that the lineage of, the isolate of a pathogen can be important because different isolates tend to have different levels of virulence. So here we have tested two different isolates. So 1613A is the isolate that we've used for all of the inoculations in the prior two slides, and here we're comparing it against CT8. So both of these isolates were collected in the Galway forest, but in 2 different years. And there's a significant difference in lesion development between the two isolates. So you can see CT8 has significantly longer lesion developments across all the clones it was tested. But interestingly, there's similar lesion development within the clones. So if you look on the left, R0408 has shorter lesions no matter which isolate you inoculate it for, which suggests that any kind of resistance that we might find may be robust between different lineages, different isolates. However, again, these are two isolates from very close locations, and in future we want to include isolates from across the range that we have in the UK and also across the different lineages that we have, so EU1, EU2 and the North American lineages. Again, we also are using all excise material here, and in future we would like to move on to using entire plants. Some more future work that we have planned is we've planted out the clones from 2015. So we had a restock site that had been growing larch that had been felled due to remorum infection, and we've planted out these clones in the hope that planting them out in an area with a high level of inoculation will result in them getting infected or not infected if there is resistance. So this is a field trial, and in the summer coming we're going to go out and we're going to do records of how well they're surviving and any symptoms that we can see of infection within the trees. We also have a second site planted out in a location with no remorum present, where we've planted out both the 2015 and the 2020 trials, and these are laid out in a randomised block design which would allow us to test field performance and also could be used in future for a clone bank and the possibility of a seed orchard. The ones that are planted out here, despite being quite young, have already started developing clones, sorry, not cones, cones, which could be used for future work. We haven't done any seed work yet, but that is a thing we are planning in the future. So I would like to conclude with the fact that this is the first study of variation to remorum in Japanese larch. So again, it's performing a basis for future work, and that we do have a continuation in lesion development. So we have these trees that we're very excited about that have shorter lesions than the rest of them. And we also like to suggest that survivor trees are a good resource for further work because they do allow us to identify individuals that have some level of tolerance or resistance. And as I've already said, further work is underway. I would like to thank our funders, so Scottish Forestry, Forestry and Land Scotland, the Scottish Forestry Trust and the University of Oxford. And I think we're going for questions later on. So, thank you.
Okay, and now we will continue with Ben Bultner talking about ash dieback, and as I understand, this is connected to Francisca's poster talk a few minutes before. Please.
Yeah, hello, good morning. Yes, I can save introduction of the institute. And I just show you.
Try it. It's not working? Yes.
Yeah. I have a very short introduction, and then I have actually two parts. I will talk about a joint project called Flux for Future in the first part, and in the second part I will talk about the specific project we at the Tree Institute work about. And also the introduction I can do very short because we have seen it basically already, and I just want to go on here. So we have an idea that the plus trees, the trees that are there, that are still there, can be planted, can be selected and planted into seed orchards. And there have been several research initiatives in Germany a few years ago, but you have to understand, in Germany we have a federal system, and the state, the forest is administrated by the federal states themselves. So they have forest administrations, and they also have research institutes that are distributed throughout Germany, and so there have been local activities, and you have a problem with ash is that actually it was supposed to be used as a tree for forest transformation. Forest transformation is to be to make forests to cope better with climate change, and so it was very bad that that actually we lose this tree species. Yeah, and we have a fund for forest and climate. And this is financed by the German government, by the Ministry of Agriculture and the Ministry of Environmental Protection. And they gave a mandate to a funding agency, called the Agency for Renewable Resources, to set up and start a very large research program, actually, you can say it. And this program is Flux for Future, and it's not just one project, and it's even not just one joint project, but just a group of joint projects. They are all together 4 of them. The first one here is about monitoring. Both by remote control, but also remote sensing, but also by plots that are distributed throughout Germany. Then we have a joint project here on the pathology of a fungus, Hymenus fraxineus, and it's also on the genetics and also about the infection process itself. Then we have here the joint project FraxGen, that is headed by the Tree Institute, and we have a project here called Frax Silver. This is mainly about silviculture and also natural regeneration. And all above that is the coordination. And here you can see the structure of a joint project FraxGen. And we have on the right side, you see the 7 topics that we deal with. And we have 7 research institutions that work with it. But it's not one-to-one distributed, but it's a network. And very important in that network are here these 4 institutions, because they are the research institutes of a federal states, the forest administration. Here, this is for Bavaria. This is responsible for Baden-Württemberg. Here for the Northwest. So as 4 states actually in Germany: Lower Saxony, Hesse, Thuringia, Hesse, not Thuringia, not Lower Saxony and Schleswig-Holstein. And here is the institute for from Sachsen-Forst, from Saxony. And you see, we have three projects that are managed by several partners. So this is here the plus trees. This is the core of our whole initiative, of our whole fraction, because we select the plus trees in the whole of Germany. Then the second thing here is in vitro culture. In vitro culture is of ash. Ash is very recalcitrant, and so actually all these are all laboratories here, all the 4 laboratories that do work on in vitro culture of forestries in Germany, and so they all together try to get the in vitro culture working. And here we have a population genetics, also as a topic for several institutes, population genetics of the plus trees, but also of the monitoring sites. And we will see later, we will hear the introduction to the topic by Gregor, and he will introduce to that topic. And then we have also 4 single projects that are just managed by a single institute. And one of those here is the project on genetic architecture that I soon will be talk about. But I just want to say a few words about the plus tree selection. So you see here some spots where we don't select plus trees. These are just the federal state forest administrations that didn't join the project. And here, this is actually the area of Res Esche, what Franziska talked about. Here already all the plus trees are selected. And there are. We tried again to look at some more. We have selected them all. What's a very important basis is a published key. It is in a PDF version, but we also have a print version, and everybody uses it in Germany. So we all use the same method to evaluate the damage that the fungus does on ash. This is a big advantage over the things that we did before. And was published by Sandra Peters, Ralf Ketzel, and Gilda Langer, and they are from the projects from Frax Mon and Frax Pathology, monitoring and pathology. And by now, we have several thousand grafts from the spring grafting campaign this year. And the success is from 50 to 90%, depends on the partner. And in the moment, we test them by isolate tests, here directly on the stem. And here by ascospore tests, putting ascospores on the leaves and more. It is similar, more similar to the natural infection process. So now, I come to the genetic architecture of resistance. This is a special project of the Thünen Institute. And we want to go onto the genomics of ash. And I know there's a lot of work done already. And with transcriptomics, with genomics. But we still think it's worth to do another approach, because we think that QT analysis can be very valuable, because the statistic power is much higher with lower numbers when you work with GWAS or similar approaches. And you simply need a full SIP progeny, a crossing you need, and you need in the offspring, you need variation of a phenotype. This is what you want to observe. And then the problem is that it is hard to cross ash in the field, and that's why we have an approach to collect the seeds from single tree progenies, doing genotyping with microsatellite PCR, and then and that we take advantage for this breeding river crossing approach from the work that we did in the project Ras Esche. We could select from all the selected trees 5 mother trees that are distributed over a range of 200 kilometers from west to east. The seed harvest was 2018. They germinated in 2019, grew in pots in 2019 and 20, and then we established a field trial that grew last year, this year. And altogether, we have made a microsatellite analysis of 4,300 progeny around from 5 mother plants. We used 8 microsatellite markers, and we did a parent test with colony. And what came out was first that the number of full-sip progenies, of full-sip families, the membership number of full-sip progenies was relatively low, below 100. We expected a little bit more because we tested 1,000, and but we went on. 950 of this progeny, we did an Illumina resequencing from 4 mother plants because one of the mother plants just had full families with 17 and 23 members. And you see the difference with the markers. So we have 8 markers and we have 1,400,000 markers. And we have a principal component analysis. And what you see here is we had by colony, we have here the mother, and we have here actually four fathers, and it turned out that they collapsed just to single one. So this spot here actually are 200 samples. We had 200 plants, and it turned out that it's actually a large full-sip family. And go on. So similar you can see here, actually 2, 4 altogether, they collapsed to 2. Next one. Here we had just one family by markers, and also one family by the colony, and here one by the snips. And go on. And the fourth, we had 3 colony families, and again 3 families by snip markers. Go on. And we constructed from that 4 large families of full siblings, each from another mother tree, and we think that the numbers here actually are now large enough to do the analysis. Now is the last slide. And now we have to do phenotyping, or we are in the process of doing the phenotyping. So here, next spring comes the ash target that we are actually interested in, the main interest. The bud growth is already done. The chlorophyll index is in progress. Here's the shoot growth. 2021 has been done, and we can do it for 2022 soon. And the sampling for phenolic compounds has been done in 2022, and this analysis in the laboratory is in progress. And then I'm open for questions, and I thank you for your attention.
Thank you, Ben, for a really interesting talk. Luis has now changed the schedule. So we are well ahead of time. And then we decided that Rosa will give her talk before lunch. And then we will see what the time is. And likely we will start the session after lunch with questions. And then we will ask Jeff, Ben, and Rosa, and Heather to come up and answer questions.
Sin miedo.
Okay, thank
So
you.
please, Rosa.
Thank you, Rosa, for moving your presentation. It's instead of some of the colleagues from Korea.
Don't worry. Okay, good morning, everyone. I don't know. Can you hear me well? Now? Okay, thank you. So, I'm Rosa Sanchez. I'm a postdoc at the University of Birmingham. I'm studying the effect of the elevated CO2 concentration and how impact on the growth, the photosynthesis, and also the defense on oak seedlings. So, First, we are studying oaks, as is one of the most representative species on the European forest, principally Quercus robur and Petrio. However, these two species are endangered by climate change and also some pathogens. The most important are the oak powdery mildew, that is a fungi that cover with a white mycelium the leaves, and the acute of decline, that is a syndrome. It's not just the 3 genes of bacteria. The tree needs to be weaker due to drought, for example. With this presentation, I will present our results with the oak powdery mildew. So, related with the elevated CO2, we know that this is one of the factors associated to the climate change that can affect the plant interaction inside the plant with the photosynthesis and growth, and also all the other interaction with herbivores, the predators, and also the pathogens. So just to try to see the effect of this elevated CO2 on the pathogen, the oak powdery mildew, we use, in culture chambers, we grow the seedlings under ambient and elevated CO2, really high concentration of elevated CO2, as you can see here. And the first response of the seedlings was a faster and early emergency of the seedlings, around 1 week before. However, after this week, more or less, the seedlings have had the same final growth. Also, with the powdery mildew, when the seedlings had around 1 month old, we infected with a solution of the spores, and also we can see the enhanced susceptibility to the powdery mildew with more mycelium covering the leaves, and also a number of the colonies per leaf. So if the elevated CO2 causes enhanced growth but more susceptible, maybe we have here a growth defense trade-off. In order to see this trade-off, we treated these oak seedlings first with BABA, beta-aminobutyric acid. It's a protein amino acid that causes other crops resistant to a broad range of pathogens and also abiotic stresses. So with the help of Jamie, an undergraduate student, we selected the concentration of 5 millimolars of BABA that cause complete resistance without any colony of powdery mildew. In this case, we can see that BABA causes slightly growth of the reaction. It's not significant, but it's also the time. We are working with 1 month old oak, so in a species that have hundreds of years, maybe we haven't yet at the time just to have this reduction of the growth due to the BABA. So our next step was make a metabolomic analysis using both factors, elevated CO2 and BABA treatment at the same time, using BABA as a resistant phenotype. We collected the leaves at different time points, 0, 2, and 4. We have different collections. One for the metabolomic analysis, a really early time points, 0, 1, and 2 days post inoculation. And also we collected tissue for callus staining, as BABA can trigger the callus deposition. This callus deposition was studied at 0, 2, 6, and 9 days post inoculation. This is our experimental system. We inoculate this plant with first, we treat with the BABA or water 1 week before the infection with the powdery mildew. And then we treat with the powdery mildew by spraying. With these seedlings, we made physiological, morphological analysis, biomass determination, and root traits. In this image, we can see the inoculated plants with the non-inoculated plants, our MOGs, with ambient and elevated CO2. And here we have the BABA treatment 1 week before and the control of the BABA treatment that was water. So it's not a well image because apparently the water plants here have more leaves than the BABA ones. However, when we count the lost leaf with the water treatment, we can see that the seedlings lost completely the leaves. Like here, the BABA treated plants, all the leaves are on the plant. In this case, comparing the elevated and the ambient CO2, we could see that BABA protects against powdery mildew, but this effect was weaker under elevated CO2, causing also a greater defoliation under elevated CO2, because the elevated CO2 is like the severity and the disease develop faster. So we got like the last step of the disease with the elevated CO2. Here we have the physiological parameters measured with Lycor system. So the elevated CO2, as we expected, increase and enhance the photosynthesis rate. BABA presented higher values of photosynthesis rate that the water to the plants has like an intermediate value. So apparently BABA is buffering the effect of the powdery mildew. The elevated CO2 decrease also the conductance with a greater effect on the MOG plants. Maybe it's because we are treating with a really high concentration of the elevated CO2. So the photosystem are totally saturated and the stumps are closing because they have enough. And just to reduce the water loss, we have this effect. And again, the BABA-treated plants present intermediate values. Here we have the morphological values because we measure also the size of the plant, number of the leaves, and diameters. In this case, it's more or less the same. With the elevated CO2, we had bigger plants. The BABA caused a really great diameters, but not the hay. With the radicular system was the really greater effect that we observed with the BABA-treated plants, because the BABA caused like a really strong radicular system. Maybe it's also because we treated with BABA by soil drainage, so it's an amino acid, could have like an enhanced growth as an amino acid that could be related. And we couldn't see any big difference due to the powdery mildew infection. So, summary, the elevated CO2 trigger high disease susceptibility, increase photosynthetic rates, and reduce root growth. The effect of the elevated CO2 on photosynthesis are not translated into the growth. That was like a quite contrasting data. BABA had a protective effect, higher photosynthetic rates, greater stem diameters, and showed a lower height diameter ratio under ambient CO2. They also had a stronger radicular system. Under elevated CO2, BABA-induced resistance was weaker. The elevated CO2 drives differences irrespectively of the BABA condition, demonstrating that a stronger effect of the elevated CO2 than the BABA in the physiology, at least. Photostimulated as a result of enhanced photosynthesis could be invested in other plant compartments, such as defense substance against powdery mildew, or be consumed directly by the powdery mildew. That maybe is the reason that we can see the growth. In addition, we have in the by4 phase, I don't know if you know this system, is the Birmingham Institute of Forestry Research. They have the different arrays, 3 of them with fumigating CO2 to 550 ppm and 3 under ambient. Using these arrays, we are testing in under field condition this experiment with the seedlings. So here is inside these arrays, we have corridors and put the seedlings under ambient and deliver the CO2. In this case, we have also another 2 elicitors, the salicylic and jasmonic acid, together with BABA, and it's an experiment, of course, in course, because we had, like the summer 2020, that we couldn't score properly due to the lockdown, so we had the help of a technician, and we are repeating these ones. Our previous result of this summer 2020 are in this picture with the control, that means the water-treated plants, BABA, salicylic and jasmonic acid, under ambient and under elevated. Again, we could see that the elevated CO2 make the BABA effect weaker. However, with the salicylic and jasmonic acid, we have like a contrasting phenotypes. There are causing more resistance with elevated CO2, maybe due to some cross-talking between salicylic acid because they are antagonists. The scale represents between the healthy leaves and with the tissue collapse. Finally, thanks to my research group, especially my supervisor, Sturia Luna Diez, all the by4 technician support, and the different students that are employed in this project. Of course, thanks for the attention.
Thank you very much. You or I? So we now have time for the question session. So there are 4 speakers that I would like to come up. You should stay. Heather, Ben, and then Geoff.
Just again, to say thank you to Anna. The talk from Anna is tomorrow was more a kind of environment, physiology, and whatever. Today is more about breathing. Thank you, Anna.
You fit anyway, I think. Fantastic. So remember what Louis asked in the morning, that when you pose a question, please also state your name so that we know who's asking. Basically. I think I'm going to hand 2 microphones to you guys. Try not to get them too close to each other, because then we're all going to be... And then I will run around with a microphone. Yes?
Hello again. My name is David. I mentioned it before, actually, but my question is about Phytophthora, and the question I have is closer. Okay. So, did you work on different types of other species of Phytophthora other than Cinnamomum, like Rumorum or other species? And about the trees, did you work on different types of trees other than larch? Do you have problem with that?
No.
It's correct.
Does that work?
Now it works. Now it works. Leave that one before
This
you
one?
go close to this one.
Does this one work? No?
It works with Louise, so you just have to speak louder.
I just have to speak louder. Sorry, what was the question? My top the Something about phytophthora.
So, yeah, I was wondering if you work on different other species of Phytophthora other than Cinnamomum, and about the species of trees other than larch. Did you, and you know, because there are other species that we have problem with in North America, so I wanted to see if there is somewhere.
So this work was Phytophthora morum on larch. We've also done work at Forest Research previously on juniper. This doesn't work, I'm just going to shout at you. On juniper and Phytophthora, oh Jesus, sorry. Now it works. On Phytophthora ostracedri and juniper. We're also doing work on Phytophthora ostracedri and Lawson cypress, because I know in North America you've worked on getting Lawson cypress to be resistant to Phytophthora lateralis. So we were looking to see if the same stuff has resistance to Phytophthora ostracedri. But we haven't finished that work yet, so I'm not talking about it today. So it's mostly just larch and morum that we finished the work for. There is a published paper on ostracedri and juniper, which was showing there is some resistance in juniper to ostracedri.
Yeah, mainly the main reason I asked the question was because to see where are the common area that you guys are working. Because Cinnamomum is not that big a deal for us there. Rumorum is more problematic in Pacific Northwest. It causes lots of problem in Washington, BC. In BC less, but more Washington. We are very worried about that one actually.
I think
coming
for
that
you
way.
guys, rumorum is on oak, is it not?
Sorry? I can't.
Tan oak. So yeah. So we have it on a conifer, you have it on a broadleaf. We're not entirely sure what the difference is and why it's decided to switch. But if you guys get it on your larches, that will be interesting. Sorry.
We have some
It'll be very sad.
Actually, we are seeing some problems with pines. Some pines are showing Phytophthora problems. Actually, Richard is working. Sinesco, he found some resistant. He did a great job, actually. I should mention that, and I'm going to talk to him about his results. So he's a very good source, actually. If you can connect with him, Richard Sinesco in Oregon. He worked on Phytophthora on pine, and he has an extensive, basically, research program on that. Thank you.
Well, this is a question. My name is Jonas Oliva. I work at the University of Lleida, and this is a question. This powdery mildew experiment with the elevated CO2. And you mentioned that you found higher susceptibility, and you linked that with higher transpiration under. Do you think also that perhaps those leaves, they have a higher content of carbon, more like reserves in terms of starch, and that could benefit a biotroph like powdery mildew?
Yeah. There are a few studies also with the by4 phase. Carolina Mayoral is studying the carbon fluxes with the CO2. Obviously can have an interaction, but we can see here exactly. We think that this high susceptibility is just because the development of the disease is faster, especially the last stages that we can see in the by4 phase with the seedlings under field condition is like in chambers, due to the size of the chambers, obviously, we just have the seedlings like 14 days post-inoculation. It's a really, really short time, so we can see just the mycelium on the leaves, the tissue collapse or the loss of the leaves. With the elevated CO2, we can see this loss of the leaves, so maybe it's more related to the development of the disease.
Thank you.
Thanks. I wanted to ask about the QTL work in ash with the breeding without breeding approach. My question is, in hindsight, did you find that to be a really satisfactory way to develop your full SIB families? Would you do it again? And could you extend this on a bigger scale if you wanted to do more genomic analysis?
Naja, this was the biggest scale we had because we are restricted by funding, and so 950 was was a limit that we had from a funding agency, and so it was the largest scale that we could do. But I think actually it's worth to think about it, to do it. And I actually would also do it again, because you have also to see that it's not always easy to get genomic DNA from thousands of trees and the quality you need for genome analysis, and DNA for microsatellite study you always get. Yeah, so we had used actually quick and dirty method. So it sounds like 4,000 samples or 4,500 samples. This took a long time, but it went relatively quick. Yeah, because we used quick and dirty method, and then had we with 8 markers, which was relatively quickly done. So I can imagine to do it again, actually.
Hi. Okay. Hi, Jeff. I have a question to you. This is Marcin from University of Tennessee, and I wanted to ask you about your Argentinian pines and the Cyrex. So my question is whether there is any species of pinus that would be resistant to this insect, and whether you mentioned that the pinus hybridize so easily, whether that would be a feasible way of introducing the resistance to other species, more valuable species.
I don't know about resistance to Cyrex. This is something that I wasn't working on at all, wasn't even on my radar screen before I went to Argentina. I do know that there are certain groups of pines that are more susceptible than others. But yeah, I mean, that would be something that we'd be interested in looking at. And in terms of the hybridization, I don't know that, maybe I exaggerated. I don't know how easily they do, but I know that there are hybrids between all of those species. And they haven't been really around long enough in Argentina for there to be probably a significant amount of that, but depending on the seed source, there may be some elements of different subspecies within the populations. And I think it's a starting point to look to see if there's variation. But I don't know. I mean, I think 5-needle pines are probably less susceptible, but they don't hybridize with 3-needle pines. So.
What pines did you say were less susceptible?
I mean, I don't want to give you guys
just
bad
take it prerogative.
But Cyrex will actually attack a lot of different species of conifers. So in South America, it's even attacking Douglas fir and firs and spruces. But I think that 3- and 2-needle pines are more susceptible.
Hi, I'm Juan from Technical University of Madrid. I want to ask Heather about Phytophthora ramorum. I'm not sure if I understood well in your presentation if you rely in your inoculation method at all, or if you, for instance, have tried soil infestation, or is not used with Phytophthora ramorum?
We have tried soil infestation and not got very far with it. So in the lab we're working with Rorum and Ostracedri, and we've not managed to get either of those to work. So what we're doing is we're relying on sporangia and Zuthbor suspensions onto wounded bark, because we know that works, and that's a method we've developed that's very reliable. We do have a PhD student who's currently working on using millet seed that's been infested with Ostracedri in this case, and trying to get that to infect the roots of juniper. I'm not sure she's having much success with it, but she is just working on it at the moment. If you have a way that works, I'd be interested to find out. So, yeah.
Thank you very much. Yeah, this is Nadir Erbigan. I'm University of Alberta. This is the question for BI4 project related to CO2. Okay, you mentioned that elevated CO2 didn't yield any plant growth, right? So what happened that the extra CO2 or carbohydrates they produce? Did you look at the root biomass?
Yeah, we look at the root biomass, and it's not a difference into the weight, it's more the length of the root. We think that maybe it's some exudate, because there are a few papers that say that this excess of carbon assimilates can go through the root as an exudate of malic acid. And maybe this malic acid is burning our secondary system, so we can see these really short roots with the elevated CO2 that are not appearing with the BABA. But maybe because BABA as an amino acid is triggering some root traits.
We found actually the root exudates can change the soil nitrate composition, nitrification process. I'm not sure you guys working on that aspect. That might be very interesting to look at it.
I think it's Sami Yuta, one of the co-director of the BIFOR, that is right now studying the biomass and the microbiome with the elevated CO2.
Cool.
Hi. Okay. I have a question. Actually, I have a remark and a question. So I think, Luis, you are completely right. We should actually talk about sentinel plantations. So maybe you find a group as interested to do it. So I was contacting already for this idea a funding agency in Germany. Yeah, so the interest was limited, but in the end it failed because I had no suitable partner overseas. So I was thinking about Far East. And now is a question, could you imagine that we could establish, for instance, a sentinel plantation in the US with European species? Would be a way to do this?
You can ask Enrico that. I'm working on established plantations. Yeah, but I know there's a lot of people
thinking about establishing actually plantations with an aim to check it.
He's asking about establishing sentinel plantations of European species.
Of European species in the United States?
Oh, okay. Yeah, I do have an international project on sentinel plantings between the United States, Europe, with Sweden and Italy and China. And we have planted European species in North America. We have two sites, one in Ohio, in Columbus, and one in New Hampshire, Durham, New Hampshire. And so what was the question? I'm sorry, I was distracted for a second. Huh?
To establish more sentinel plantations with, for instance, with progeny trials, for instance.
Oh yeah. I mean that's ours is a proof of concept basically, but the idea is to convince more people that this is a good strategy to essentially conduct risk assessment, ex patria, as he was saying. Oh, we did. We are doing that. We have American species in Europe and China. It's just that it's a very small scale, and it's only 2 locations. And
I'll just jump in.
Yeah.
So the reason I wanted to contrast that with the project that I'm leading or trying to develop, which is taking advantage of the fact that exotic trees are very abundant throughout the world, and that includes plantations, but also in urban forests. And we've already learned there's already a lot of information available on the pests and pathogens that impact them. And so trying to take a deeper look at that with more information and analysis to decipher, okay, let's make some sense of what's already known in terms of the diversity of pests and pathogens on trees outside of their native range that are fully developed trees, adults, and there's at least enough of them, or they're widely distributed enough to have a likelihood of encountering the pests and pathogens that may have adapted to them or encountered them outside their native range.
Yeah, so there is a lot of information on these kinds of tree, arboreta, urban trees, ornamental trees, that like European species that are planted in North America or everywhere else, right? But
thinking about planting something with an experimental design.
yes, but
somebody can use in 100 years, for instance.
yeah, but also, even though in North America, for example, we have tons of European tree species that have been exposed to a lot of different pathogens and pests, in our own sentinel planting garden, we actually found, or at least two cases of pathogens that we are describing basically for the first time on that species, which is really weird, because I mean, it's not weird, but you would have expected that they would be known by now, right? Because of all these plantings, urban forests, and so on. So I think there's merit in a very wide approach that includes both very structured sentinel plantings and what Jeff is talking about, which is let's take advantage of all the sort of off-site exotic plants that we have in each country.
Like Sitka in the UK.
Now, okay. I'm Paul Krokene from Norway, and I have nice feedback in my right ear right now. But anyway, about sentinel plants, how can you take that one step further, I mean, towards management? Because I work with pest risk assessment, sort of part-time in Norway, and I know that this quarantine business is very rule-based. So if you find something potentially really bad in a plantation, let's say an American pine species grown in South America, can you use that, or then I assume that then you will have to do a pest risk assessment and get that species on a quarantine list before it actually will prevent import of that one in whatever plans for planting?
Paul, that's an excellent question, and I'm really glad you asked that. Two things. First is, absolutely, we need to be thinking about that. I think because of the way the world works and because of the way that trade is regulated nationally and internationally, these sentinel projects, there's a lot, we could go into all of the potential conflicts of interest and things of that nature. That said, and I think we're hoping that we can kind of shift that paradigm and get people to think more internationally about diseases and things like that. I will say that I'm not really emphasizing specifically, targeting a country and saying we want to know what's there so that we know what to stop importing or whatnot. We're thinking about this more as an opportunity to gather information. And the example of Argentina is great because we don't import any pines from Argentina anyway. And honestly, the biggest potential there that we found is something that we already have in the US. So it's really telling us what we need to be doing within our country just by studying it outside. So you can learn a lot of things from sentinel plants that don't directly impact trade between the two countries that are involved in that collaboration.
So just a comment, maybe this is a different topic, but I think the most frustrating thing about working with this quarantine business is that we all know that it doesn't really work. So the next invader is always something that was not on the list, and there's like dozens of examples. So, but still, the regulating authorities, they have their lists, and if it's not on the list, it's not a problem. And but that was just a comment.
I'd also like to just say that I would love to learn more about how things work here in Europe because I can tell it's very different. And yeah.
Yes. We will, after Enrique's question, we will take one more short question and then lunch.
Well, mine is more a follow-up comment to Paul, and it's absolutely true. So the idea of testing sentinel gardens is to show the regulatory agencies and national governments that these gardens can actually alert you to the possible dangers that you're unaware of, right? So the unknown unknowns, and you can. But then the follow-up steps are the critical ones. How do you stop these things coming in? And so it would be a matter of really ramping up to very high levels the abilities of inspection teams at the borders to actually do the inspections and do the diagnostics. And so they would have to ramp up molecular labs at major ports of entry, for example, that would allow them to detect those specific pathogens or insects. So it's just the tip of the iceberg, right? But we're hoping that demonstration of the system will catalyze these conversations with the regulatory agencies in your own country, but also in other countries, and improve things overall. That's the only, I think that's the main point.
Thanks.
And then Luis has a final question.
Thank you. I have actually two. One of them is about it's for Heather. That it's about maybe I missed a little bit in your presentation, but about the correlation between the lesion length and the survival in adult trees.
So we haven't done that part yet. So what we
Sorry?
we haven't. Sorry. We started with survivor trees, which are trees that are still alive in plantations which have suffered 99% mortality, and they're still alive, well, about 7 or 8 years after first infection. So these trees are trees that are still living in an area with a high inoculum load. And from those trees, we're testing to see if they've just survived by chance. And with presuming that trees that have shorter lesion development are going to have a better level of resistance than those that develop longer lesions, although they all come from survivor trees. And now, sorry, not picked. They've all been planted out. So we've now got them planted out in sites where there is a high inoculum load, and in the summer coming, we're going to go and check if these planted out trees are still surviving. And then we can feed that back into, is there a difference in lesion length that leads to a difference in survival when they're planted out in sites with inoculum? Does that answer it?
Yeah, I think so. So it's genetic correlation between the same clones and same material.
Yeah.
Okay, thank you. And the other question is for Rosa. It's about what do you think about that is causing that susceptible phenotype? Maybe something related with the cell wall, or I don't know. Just figuring out because why the CO2 is changing the susceptibility, and if you have any clue about.
Yeah, initially we thought about the growth defense trade-off, because obviously enhances elevated CO2 have the growth effect with all the photosynthesis issues, but in this case maybe it's because all these photoassimilates are being consumed by the powdery mildew, so directly we are feeding the fungi with the elevated CO2. With the cell wall, using the callus staining that are not here in this presentation, we saw that with the elevated CO2, the callus deposition appears like more than 4 days after that with ambient, with the ambient conditions, we can see the callus deposition around the 2nd and the 4th day post-inoculation. But under the elevated CO2, the first callus deposition appears at 9 days post-inoculation. So obviously the primary defense, that is the cell wall barrier, is appearing later with the CO2, is causing also more susceptibility because we have more days that all the spores can penetrate together with the feeding.
So with that, we would conclude this session before lunch, and I would like you to join me to thank this morning's speakers. the time it took this morning, if we are going to be on time. So when the speaker that's speaking before you is speaking, then maybe you can slowly try to make your way forward in the wings for one way or the other. Så. So now, during this poster speed session, we have 5 talks, and we will begin with another talk from this program in Germany that has to do with ash dieback, and it's Gregor that's starting when we get the presentation up and running.
Wait a minute, please. Yo creo que el ratón se queda en la pantalla. Pues alguna, pero sí, te lo puse ahí.
And of course, questions for poster talks happens at the posters during coffee break.
No. So, again. Hello everybody, my name is Gregor Kunert. I'm from the FVR, the Forest Research Center of Baden-Württemberg in Freiburg in the southwest of Germany. I'm also working like Ben and Franziska in the Frugs for Future Frugs Gain project on ash. My topic is population genetics of common ash in Germany with regard to ash dieback.
Sorry.
Ben, give already.
Try again. Okay.
Thanks. Ben, give already a nice introduction to the project. Our part in the project is to genotype our intensive study sites of the project. We've got 14 you see on the right-hand side, and yeah, we are genotyping them with 12 core microsatellites and 8 chloroplast ones. These sites are fenced mixed forest stands with a high proportion of ash. Yeah, we've sampled some 21, and this year 50 adult trees and 50 saplings from natural regeneration there. And this year we also sampled again 30 adult trees for further SNP analysis there. The genotyping we perform here is also happening in the project from other partners for the plus tree selection. Ash dieback severity on these plots you can see on the right-hand side in the bar plot diagram. It's taken every summer and winter in 6 damage classes, and what we mainly see here that only 3.5% are in class 0 to 1, so the good trees are very rare, and the bigger part, over 70%, are in the very damaged class 3 to 5, where 5 is already dead. So, yeah.
Sorry.
Next one, please. Yeah, thanks. We are not much into results now because we just finished 2 out of 3 core multiplexes. So it's just a few in the future. What we've done so far at the right-hand side and the PCOA, we can see that the plots ST1 and BB1 shows quite a genetic distance to the rest of our intensive study sites, but I have also to say, when we plot these for individuals, we see that's individuals from these plots grouping with the rest. So this might be a hint for maybe some planting, or we don't know exactly yet. We will have a closer look for this in the future. Yes. And yeah, what's coming up for us? We will finish our analyzing with the core and chloroplast microsatellites, and then can go deeply into these data analysis. And now we are preparing our samples for further SNP analysis with already known SNPs. Yes, and that's all from me. Thank you very much.
And the next speaker is you, Rafa, is that correct?
Me?
Yeah, I think so.
All right.
Just something for clarification. The turn for the questions to the posters, the idea is to present all the posters today, and we have time tomorrow and the following days in the coffee break for visiting the posters on site. Okay, so just a brief presentation today and time for questions tomorrow and the following days.
Okay, hello everybody. I am Rafa Zas from the Misión Biológica de Galicia, the hosting institution of this workshop. But I am supposed to be also an organizer of this workshop, but I have to say that I didn't do anything, and Luis did everything. So I would like to thank Luis for a very strong effort that he paid. Well, I have a poster here about the resin flow in maritime pine, about the source of genetic variation and environmental variation in resin flow, which all of us know that resin is the main resistant mechanism of pine trees against pests and pathogens, but also is a valuable bioproduct that is gaining interest in many industrial processes, and that was very important as a non-timber forest product in Spain time ago, but nowadays is gaining attention again, and because it can substitute petroleum derivatives in many industrial processes, and it also has a lot of benefits for the forest owners, such as complementary incomes for forest plantations, but also many valuable ecosystem forest service, and that's, it's not only a defense, it's also a bioproduct. And in this poster, we just summarize several works done in common garden tests established in Spain, provenance and progeny trials, where we just evaluated as a phenotypic trait the resin flow and explored the among population variation across populations within the distribution area of maritime pine, and also the within population genetic variation to have a look to the reachability of this important trait, and also about plasticity across environmental variation, both in constitutive resin flow and in induced resin flow after different stimulation processes. We use a very simple way to measure resin flow, which is we make very little holes in the trunk, and we collect the resin flowing from these ones in a short period of time, usually 24 or 48 hours. And in previous studies, we have comprobated that this estimation of resin flow, this fast estimation of resin flow, is highly correlated with the resin yield that one can obtain from the trees after one whole year campaign of resin tapping. So, well, I'm not going to try to go into details of the results. The main conclusions of the poster of this, which is a compilation of different studies, is that there was very large variation, genetic variation, both among and within population in the constitutive resin flow, which was also highly plastic to environmental variation. But we didn't find evidence of genetic variation in the inducibility of resin flow after stimulation. And it was also, we found little plasticity of the inducibility of resin flow across environments. And that's it. Just more details, we can see them in the poster. Thank you.
And then we have a talk from Vilnius Tree-Pars on promoters in pine.
This one.
Vilnius. Número 10.
No. Suppose.
Ese sí es este.
Okay.
Hello, my name is Vilnius Tree-Pars. I am from Latvia, from the Latvian State Forest Research Institute Silava. And my scientific supervisors are Dainis Rundis from Silava and Adam Vivian Smith from Nibio in Norway. So a short introduction about what happened before the current research is that I made an inoculation experiment to find some candidate genes for Scots pine resistance against root rot caused by Heterobasidium annosum. And once we found those candidate genes, the next question for us was how are these genes regulated and what's further upstream from the genes in the genome. And of course, we don't have the reference genome for Scots pine yet. I know that people in Sweden are working on that, but it's still not published. And this makes it hard to design primers to study what comes before a known sequence. So I used a transcriptome of Scots pine from a previous study, not performed by me, but the details are on the poster, to create primers and to use them in linear DNA amplification. And later, after the linear DNA amplification, the goal is to sequence what we amplified. And this method here was basically, I don't know if I can say that invented by me. I chose this protocol to do it. So the first step, of course, is to get very high quality, high molecular weight DNA because for a lot of reasons, including that we would later use Oxford nanopore sequencing. And the most difficult thing here is the circularization of single-stranded DNA. This is already stressed by the manufacturer of these reagents, together with whom I worked to get a better protocol to be able to create long circular DNA molecules. After you get the circular DNA molecules, you treat the sample with the DNases, which degrade all the rest of DNA except the circular single-stranded DNA. After that, we performed rolling circle amplification, so all the controls and everything looked good until that point, and actually I was a little bit surprised about that, because it's a very, very sensitive method. If you have some sort of contamination, then it automatically bad. And after the rolling circle amplification, we sequenced the product by using ion torrent sequencing, together with a specific kit from Swift Biosciences, which allows you to use single-stranded DNA for library preparation. And the other technology was, after additional amplification with hexamers or specific primers, Oxford nanopore sequencing. And the additional amplification is necessary because it cannot use single-stranded DNA as the source for library preparation. And in the end, 2 out of 100 selected candidate genes which I targeted could be found in those next-generation sequencing data. And basically it's 2% efficiency. So I'm very happy about it because it worked, but I'm not happy about it because it works so poorly. This is a postdoc project, so I have limited time and limited money. So we decided that I have to switch the method to something better. And I substituted this method with something based on terminal transfer amplification and sequencing method, which is also very fine and interesting in my opinion. So the start is the same. We make nice high molecular weight DNA. Then there is not multiplex, but singleplex linear DNA amplification, which the products of which you shouldn't be able to see. If you see something in the gel, this means that there is something interesting either in the or structure of the genome, or you have non-specific amplification. And then this linear DNA product was treated with the terminal transfer amplification method, and sequencing was performed by Oxford nanopore sequencing. And we found, as a result, all the primer binding sites which we did intend to find, but just next to the primer binding site was something totally else from Scott's pine, but not what we expected from the transcriptome data we based our primer design on. So we designed the primers in a way that there is a short piece of sequence from the transcriptome after the primer, so we can check it, whether it is what we expect to see or not. So our primary suspicion was that there is additional places where the primers bind, or that there are some mutations in these places. So we performed whole genome sequencing to clarify what's going on. We used the Q20 reagents and Oxford nanopore sequencing, and just a small portion of the reads, so less than 900,000 reads, showed us that we can find the primer binding sites and the transcripts in this small genome, and only 2 of the target sequences were not found. And from this material I can also get the additional sequence before the candidate gene. My poster is there next to the window, and I am here, so any questions after that? These are my scientific supervisors. I thank them for letting me try something this complicated. Thank you.
Thank you very much. So our next speaker is Barley Rose Collier Harris, it was. Sorry very much. And you're going to talk about Sitka spruce and Hylobius, right?
Yes.
Excellent.
So I'm talking about an investigation of naturally occurring defense mechanisms in Sitka spruce to inform research into Hylobius aviatus resistance. Oh, and I should say it's a mixture of my work and Haley Tumas and Zayad Sufi's work, who are 2 postdocs in my lab before. So Heather briefly talked about Sitka earlier to say that we plant a lot of it in the UK. It's the 3rd biggest crop in the UK by land area. That includes all arable crops, so it takes up a huge amount of space, and it's about £1 billion industry per year. But all of the Sitka that's grown in the UK is from one provenance, so from one area in Canada, and we've got a breeding population in the UK that's been selected for growth traits. Hylobius, the large pine weevil, is a native generalist pest, and it eats bark. And the reason that it's a problem for Sitka is that it eats the bark, it girdles the stems, and if we don't spray with pesticides, you've got on average 60% mortality and up to 100% mortality. So what Haley and Zayad did was that they extracted terpenes from 50 full-sib families and compared the amount of terpenes in the full sib families in the UK to populations from Canada which are known to be resistant to the white pine weevil, a different weevil, or susceptible to the white pine weevil. And you can see that for terpinolene and for 3-caline, you've got pretty low terpene concentrations compared with the both susceptible and resistant Canadian populations, but that we've got a pretty similar concentration of dihydroic acid. Yeah. So I then looked at some of the families in the UK breeding population and did a damage response study where they got attacked, controlled exposure, 72 hours of attack, and we got high levels of attack in all of the population, and we saw no differences between family, which could be because they're all from this similar area, so there might not be that much variation. So following on from that, we've got kind of 3 main plans. So I'm doing a field trial and a greenhouse experiment looking at damage and amounts of damage that are done, but here using provenance material that's been sent. Seeds have been sent over from all different provenances across the native range, and I'm growing them up, but that's taking time, obviously. And we're also going to be comparing some full sib crosses from Canada which are known to be susceptible and resistant to the white pine weevil and see how Hylobius attacks them. And I'm doing an immune response study to see how Sitka responds to Hylobius and attack, because I hope that at least something will be statistically significant in my PhD. So that's what I'm up to. My poster's over there. Thank you. My sponsors, my website.
And the final presentation for this poster speed talk session is Sarah Bogevs that's going to talk. Oh God. That's probably a good space. Yes. Brilliant. That's going to talk about pathogen detection, if I remember correctly. Are you in a good space? Yes. Fantastic.
We'll see. Hi, everybody. So we took 2 diseases that we frequently work with, thousand canker disease and laurel wilt, and developed a molecular detection system for those using TACMAN probes. So we tried to develop something a little bit quicker and easier to use. Previous methods of detection either took multiple weeks to grow out pathogens and try to isolate those individually, and we also had developed a different molecular detection program with TCD, but thousand cankers disease. But that one was a little bit more expensive, so we wanted something cheaper and easier for people to utilize. So we started to develop a new detection system, which we are hoping to be able to use with other disease complexes as well, that uses TACMAN probes. And we're able to actually use TACMAN probes with a normal gel electrophoresis. We're able to use a qPCR machine to visualize the results. And we're also able to use a specialized flashlight that is pretty inexpensive and some glasses to actually visualize that light underneath UV. And that will allow people specifically using microsatellites to specifically see the pathogen and know that we have that pathogen present. This method is much quicker, it's cheaper, and we're hoping to be able to let diagnostic clinics be able to use it easier and then use this for other insect vectored pathogens to be able to confirm quicker. And it has been a huge combination of people to work on this, and hopefully we'll be able to work on it with other pathogens and see how it works with others and use it for quarantine measures.
That was it for this poster session, so I would like to thank all the speakers. And maybe let's see, either I should hand over the word to you, Kate, or I should, Luis.
All right. Let's go ahead with the program. I'm Katerina Villari from the University of Georgia, and we will have a group of 3 speakers now, question at the end, and then coffee break, which I know you're all looking forward for, always. All right. I think the first one is Raquel. Raquel, is that correct? Or okay, good. I never know if I get the name right. Raquel, I will show notes, and then if you're really, really late, I'll stand up and start dancing beside you, so you know you really have to leave.
I would try.
Okay, thank you.
I'm sorry, but I think it's better to be like this.
If you want, you can use the
Be there then? Yeah. Yeah, for me it's much better to be here. Well, I'm Raquel again. I'm going to talk this time about the variation in susceptibility to pine pitch canker among 33 provenances of Pinus pinaster. This work was done in collaboration with a group of Julio Díez Casero from the Valladolid University. Well, the Fusarium circinatum is an ascomycet that causes the pine pitch canker disease, and it affects at least 57 species of Pinus, but also it affects Pseudotsuga menziensis. The mountain pine, Pinus radiata, is the most affected species. And this pathogen has been declared as a quarantine pest by the EPO, and it has been included in the A2 list. Well, what are the symptoms? The symptoms are different depending if it attacks plants in nurseries or adult trees. You can see that plants in nursery usually have damping off and then they die, but some seedlings are asymptomatic. In adult trees, you can see needles turning into yellow, then red, and then they drop. But you also can see crown dieback or cankers with resin exudation. Here you can see a photograph from the north of Spain with this disease in radiata pine. You can see that almost all the trees are dead. Okay, what's the dispersion? You can see that it is almost all over the world. Here are the countries where it has appeared at least once. It appears for the first time in North America in 1945, and in Europe the first detection was in 2005 in Spain. In the map on your right, you can see the spots we have or we had in 2015 in Spain, in the north of Spain. Okay, the vectors which disperse this ascomycet are human vectors, insect vectors, or even the wind. But we also have to be very careful with infected seeds because they are going to produce, in most of the cases, infected seedlings, and they are going to go into plantations. So we have to be very careful with the control measurement methods in nurseries. Which are the control methods we have right now? Oh, okay. About management, we can do heat treatment of seeds. We can disinfect tools and sterilization of containers, pruning infected materials, and control the insect vectors. But we also have biological control, such as endophytic fungi, microviruses or RNAi. And of course, we have genetic breeding, looking for resistant genotype, which is the thing we do at our center. Talking about the host, Pinus radiata is a circum-Mediterranean species. You can see that it almost is in Portugal. Spain is mainly in the north part of Spain, southwest of France, and northwest of Italy. In the map on your right, you can see in red the provenance regions, in green the natural stands of Pinus pinaster, and in blue the plantations. You can see that here, the place we are in Galicia, there are a lot of plantations. So it's a very important species for us. In fact, for the industry, it's really important. Almost 1.6 million of cubic meters were cut in 2017. For this reason, we have a breeding program of this species. We wanted to study the effect of this fusarium, of this disease, on the species and then on our breeding population. But in this case, we are going to talk just about provenances. Okay, in this study, we use 6-month-old plants. We include 33 provenances, the 28 from Spain and 5 from abroad, 2 from Morocco, 1 from Portugal, 1 from Italy, and 1 from France, Corsica. And then we also include 3 seed lots, 1 from the French second generation breeding population, 1 which was our breeding population, and then a radiata seed lot, Pinus radiata seed lot. Okay, the questions we wanted to answer were, are there significant differences among provenances? Is there any correlation between growth and Fusarium circinatum resistance at population level? And if so, is the Fusarium circinatum resistance still significantly different among provenances when including growth as a covariate? And just more practical, the last one is a more practical question. What is the resistant level of the Galician breeding population, which is a control we use, compared to the resistant level of the Galician provenances and the French breeding population, which was used as a control also? Okay, we did inoculation assay in collaboration, as I said, with the Valladolid University, so they did the work there. We used a randomized complete block design with 2 treatments, 4 blocks, 36 genotypes, the 33 provenances and the 3 controls, and control seedlots, and 4 seedlings per plot. The damage scale was a 4-level scale from 1 to 4, 1 completely healthy plant and 4 completely dead. And here you can see a graphic with the mortality evolution through time. There are some highly susceptible provenances. In fact, all the seedlings died, while there were other provenances which have less than 50% of mortality. Analyzing data, we saw that there were significant differences among provenances. For this analysis, we just used all pinaster. We didn't include the radiata control, but then for this representation, this graphic, we did the analysis again, and we included radiata pine just to check the level of damage of the others. So we can see that there are some good provenances and others, such as PT233 and PT223, which have the same level of susceptibility to the pitch canker. So there are significant differences among populations for resistance to this disease. So the first question is answered. But now we wanted to see if the growth had any relationship with this. So we analyzed initial diameter and initial height for different provenances, and we saw that there were significant differences for both threats. So we analyzed the correlation between the blobs of both of them with the survival, and we saw that there were no significant correlation. So it seems that growth is not related with the disease resistance. So we can still say that there are significant differences among provenances for this threat, for this disease, but it's not related to the growth. Here in this map, we have tried to represent the worst provenances, these two in red, and the best ones. The last question we wanted to answer was how was the level of resistance of our breeding population compared to the Galician ones and to the French breeding population, because for practical reasons, maybe are the 4 seedlots that could be used here in Galicia in plantations. We repeat the analysis just with these 4 populations, so they were considered as fixed factors. And we can see that the coastal Galicia provenance and the Galician breeding population were slightly significantly more resistant than the inland Galicia provenance and the French breeding population. So this should be considered when planting, when establishing new plantation. Of course, it's slightly significant. It's not for sure, but for this reason, we consider that we have to do more analysis, more essays, to check if this is confirmed. So we should do any other essay with a bigger sample size. So as a conclusion, we can say that there are significant differences among Pinus pinaster provenances in resistance to pine pitch canker. No negative genetic correlation was found between survival and growth. So it seems that there is no trade-off between these both threats. And even though only slightly significant differences were found between coastal Galician provenances and Galician breeding program on one hand, and inland Galician provenance and French breeding population on the other, we should be careful using the least resistant provenances or seedlots. And that's all. Thank you very much.
Thank you. So we'll go for question later on. So I'm calling the next speaker. I think it's Rahul or Rafa. Are you presenting this? Or Rahul? Okay. Rahul de la Mata, and he's going to talk about effects of the genetics and the application of methyl jasmonate and Hylonox protector on tree resistance to the pine weevil in 2 widespread boreal conifer species. Rahul, the rules are the same. I'll show you notes and stand up.
Yeah,
thank you.
Which one?
Which one? La única que tengo.
Workshop Tree-Parasite. This one.
Sí.
Okay. Thank you very much.
Okay. Thank you. First of all, a little bit of leadership, funding, and these kind of things. This is part of a project. It's a collaborative project between the Swedish University of Agricultural Science, SLU. The leader is Adriana Puentes, that is not here, but there's a PhD student, Cristina, that is here from this group. And it's a collaboration with the group of Rafa and Luis. Luis is the leader from this project from the CSIC, and I've been working with them last summer in Sweden. So what I'm going to talk today is about Norway spruce and interaction with the pine weevil, Hylobius avietis. This is funded by Formas from the Swedish government. So a little bit of science philosophy. Plant-insect interactions are big drivers of plant evolution. What means that how we see plants today is because the interaction with the insects. Sometimes it's positive, like seed dispersal, like pollination. We have to take into account plants are, they cannot move, right? So they look for other organisms to move for them. Also, some insects help plants to defend themselves. But mainly today we are focused because the insects is a big pressure, right? They try to eat plants. Actually, everybody tries to eat plants. And plants, they want to defend. So since 60 years ago, in the field of ecology, focusing on plant defenses has been a very relevant topic, right? So there's two main mechanisms of plant defense. By one hand, we have the resistance. It's known as the sword, right? The plants fight back the attack. They try to prevent, stop, or reduce the damage. But there's also other mechanism that is not as well studied and has not been as deep considered. Is the tolerance. Plants try to withstand, recover from damage, boost photosynthesis, and even in Norway spruce, they can heal the wound, right? This is a different kind of mechanism. How to measure this or how to estimate these kind of things? Here we have a couple of figures, right? This is the figure for the resistance. I mean, more defenses, less damage, right? Different genotypes, for example. The most defended genotypes, they show less damage. But this is the graphic for the tolerance. Here we have two different genotypes. Under the same damage, phenotype, genotype B keep the thickness. It's about this slope, right? When the damage is increased, the thickness is kept somehow because, I mean, it's boosting the photosynthesis, it's healing this kind, the wound, right? So we have two different mechanisms, and what is relevant, they are under genetic control. Different genotypes, they have different tolerance strategies or resistance strategies, and even more relevant, they can be in trade-off because the final resilience of the plant is a combination of the tolerance and the resistance. Actually, both mechanisms, they can be using same resources, so it's expected some kind of trade-off here. But also we have two different strategies in defenses, but in terms of resistance, right? The classical constitutive induced defenses. These ones, the constitutive, they are basal, right? They are always there. And the induced, they are just after an attack or when the plant detects the attack. Again, they are under genetic control, has been shown many, many times in plants and mainly in trees. And they can be also, and we know they are in trade-offs. So this is like the basis of what I'm showing today. Also, we know that there's some hormones like the methyl jasmonate that can induce defenses, because actually are the hormones that the plant uses when detects the attack to induce defenses, right? And after induction, the plant is more defended or is at least primed. We can use this kind of hormones to increase defenses. So what we did, our study system is focused on Norway spruce, super important species. Sweden is a forestry country, 73% of the land is forest, and 80% of this forest is cultivated, right? It's long-term, long rotation, looks like natural, but actually it's not as natural as it looks like. And from this land, or the forest land, 42% is Norway spruce. So it has a very relevant pest, that this Hylobius aviaris is a weevil and attacks the regeneration. Actually, it's a silviculture pest because in usual conditions it's twigs and small branches and doesn't matter. But how it's managed the forest is more dangerous. I'm going to go a little bit fast, but I mean, after clear cutting, that is how it's managed, this kind of forest, the stumps attract adults. The adults lay the eggs. The eggs feed on the roots of the stumps. But when they rise, and even the adults, they eat the twigs and branches. But if they find recently planted seedlings, So they can do this. It doesn't matter in an adult tree, but it does matter in the seedlings, right? And they kill the seedlings, and it creates a big money loss, right? So this is why it's called a silvicultural pest. Maybe in natural forest, these weevils is not a big problem, but in these even age forests that are managed in clear cuttings, this weevil is very relevant, creating mortality, very high mortality, right? So what is done in Sweden? There's some silvicultural measures, like scarification of the soil to get out the eggs, shelter wood, and they use a lot coating or some kind of barriers, these physical defenses for the plants that they came from the nursery. Also insecticides and biocontrol. Until my knowledge, there's not a lot of biocontrols of weevils, right? But insecticides for sure, they are trying to reduce these insecticides. So how can we help to that? I mean, I talk about that. We can boost plant defenses, right? We can try to help the plant to defend itself. By one hand, the classical tree genetic improvement, we can select genotypes more defended. But also we can try to boost tree defenses using chemicals, right? I talked before about the methyl jasmonate. So what are the questions we have here? I mean, the main question is how can we help to boost tree defenses to avoid using insecticides and other expensive measures? First question, can we boost effective defense under field condition against the pine weevil applying methyl jasmonate? Is there any physiological cause by applying methyl jasmonate? I mean, it's a hormone, makes things within the tree, right? Is there a genetic variation in tree resistance? But is there in tree tolerance? And more interestingly, what's the trade-off between tolerance and resistance? How can we play with these two things? Can we increase both? It could be perfect, right? So we have just one progeny trial in southern Sweden. This is the clear cutting where this has been planted in spring 2021. This is the progeny trial. 40 elite half-seed families from the Skogfors Norway spruce breeding program has been tested in 19 complete blocks, many trees per family. They has been planted narrowly, relatively narrowly. All these trees, half an hectare. So 3 treatments of application of methyl jasmonate before planting the trees in the field. These 3 blocks. 3 blocks protected with a physical barrier that is, the industrial name is Helonox. Actually, the weevil cannot eat the plant, probably because it sands the weevil mouth somehow. And the other blocks has not been treated at all. What we measure, I'm not going to go in deep neither in the measurements, neither in the statistical analysis. If some of you are interested, you can contact me or Luis and Pedro to see how we did, right? It's just because timing. We measure growth, diameter, and height, the highest wound, the wounded area, because the weevil, of course, in different times, mainly in spring or at the end of the spring and at the end of the growing season. The weevil is eating in spring and over the summer. And also, we measure things like damage, just yes or no. These ones are not normal distributed traits. These ones are normal distributed traits. This is a binomial. Survival, of course. Not always, no. Trees can be eaten, but not always die, right? And percent of area wounded, like a ratio, also binomial. So this is Adriana Puentes applying the methyl jasmonate in a nursery. First result. I mean, wounded area. We can see, I mean, this measurement is independent of this one. We try to separate the wounds from June to the ones in September. Norway spruce heal the wound somehow, right? So we can see that over the summer, the weevil eats more. In terms of the percent of plants damaged, we can see that there's not many difference between June and September, what means that most of the plants are already tasted by the insect in June, right? In terms of survival, we can see a decrease of survival with time. Insects that ate in June caused mortality in September. And in terms of percent area wounded, we can see also an increase over the summer. So this is a comparison among the three treatments: Helonox, controls, and methyl jasmonate. So in June 21, we can see that methyl jasmonate and control, they were the same. They show the same level of wounds. But Helonox is actually effectively protecting the plants, but not the methyl jasmonate. But in September, methyl jasmonate and Helonox, they are protecting almost the same. So what is showing? That the methyl jasmonate somehow has a delay in the protection. It starts the incitation of defenses, but takes some time, right? If we consider the total wounds over the season, Helonox protects better. Methyl jasmonate protects a little bit worse, and of course controls that are not protected are showing more level of wounding. If we measure this in terms of repeated measures over time, we can see the same result, like total wound as repeated measurements. Now we are comparing the percent of seedlings damage. In May, Helonox shows less percent of seedlings damage. Controls are intermediate, and methyl jasmonate shows a lot of seedlings damage. It looks like the treatment with methyl jasmonate and all the defenses are attracting the insects, right? Really. In June, we have the same result because it's just 1 month. And finally, at the end of the season, the three treatments show the same level of seedlings damage, right? So finally, the insects, they taste all of the seedlings, right? But it's interesting how methyl jasmonate is attracting the insects to the plants. In terms of survival, Helonox is showing the best survival rate, and control in June is the same. And methyl jasmonate is increasing mortality somehow. I mean, the hormone is increasing this mortality. In September, control and methyl jasmonate shows the same level of mortality. So, and like a total measurement, control and methyl jasmonate are showing the same. What means that somehow methyl jasmonate protects the seedlings in terms of the amount of wounds, but also triggers some mortality in the plant, probably because physiological mechanisms within the seedling. Helonox is actually showing the best performance, right? And interestingly, in terms of height, methyl jasmonate is protecting the plant in terms of wounds, but is decreasing survival and is decreasing also growth, right? It has a growth cost, and here we can see it clearly. In terms of genetic variation, I mean, growth has genetic variation, mainly height. At this stage is the usual thing. And all these variables in terms of wound area, percent of damaged plants, and survival, they show some genetic variation, but as it has been shown in previous works, the genetic variation in resistance to the weevil is not very high. And finally, here we can find a genetic correlation between wound area and growth. The families, every spot is a family that grows faster, they are showing more wounds. So probably there's a trade-off between resistance and growth. So I'm going to go very fast because I'm out of time. Many of these questions I already have talked about. And as you can see, I'm not talk about the tolerance, and the tolerance will be the next step, and basically it's a correlation between the level of damage and how these seedlings recover after the damage, mainly in terms of growth, but also in terms of survival. I'm sorry because I'm going very fast in this last stage, and thank you so much.
Thank you, Raul. Very nice presentation. All right, I'll call the next speaker before the discussion and then the break. Enrico Bonello, it's all yours. And Enrico's going to talk about infrared spectroscopy and machine learning for rapid host resistance screening.
And I have half an hour.
No, you don't.
It says it goes to 4.
Go ahead.
Okay. Let's get into it. Let's see if this works. Yep. So the question for me for the day is there a way to phenotype plants for resistance to a specific pathogen in a non-destructive manner? So avoiding actual inoculations, infestations, which kind of follows nicely to a lot of the talks we've heard today about screening for resistance. So I want to talk about a few case studies where we used infrared spectroscopy to do this. The first one is coast live oak resistance to Phytophthora ramorum, which is the causal agent of sudden oak death. And sudden oak death is a very important disease on the west coast in the United States. It kills coast live oak, tan oak especially, and also California bay laurel. And it's actually a kind of a syndrome because there's other players involved in the mortality of the trees, like bark and ambrosia beetles and various secondary fungi. So in work that we started very early on, back in 2002 actually, we found that with coast live oak, some trees displayed very high levels of resistance, which was evidenced by very small lesions in response to artificial inoculations in the field. I'm talking about field-growing trees, wild trees, that we were able to inoculate on the stem with Phytophthora ramorum. And it turns out that we found a good association between certain phenolic compounds that can serve as biomarkers for this resistance. And but the immediate question was there must be a faster way to do this using HPLC, right? To phenotype trees for resistance using constitutive phenolics, I should say, so before any infection ever occurs. So how about infrared spectroscopy? And this is where I have my little dancing molecules. They're a little sluggish today. This is just to show that infrared spectroscopy actually elicits a variety of agitation in any molecule that they hit. And this is highly dependent on the specific bonds in specific molecules. This is just a methane molecule. And, each of one of these 4 different basic or foundational vibration states absorbs a different wavelengths or wave bands as are expressed here. So if you can imagine, I'm explaining this for those of you who are not very familiar, but if you take a mixture of compounds, you can create a spectrum based on all these absorbances that is the result of the sum of all the absorbances of all the different molecules and all the different bonds in those molecules that you have in a mixture, right? So you can do that with extracts from plants, basically. And so most of the talk today will be about Fourier transform infrared spectroscopy. And this is a technique that actually uses mid-range infrared radiation. And the way you do it, you put a sample on a glass slide, and then you bombard it with an IR beam, and you get an absorption spectrum out of it. So the idea here is that this vibrational spectroscopy gives you a snapshot of the overall chemical composition of a given tissue at any given time that we call chemical fingerprint. And then we can use statistical tools or, as I will say a little later, artificial intelligence approaches to classify trees into resistant and susceptible based on their chemical composition. So I should say the assumption upstream of all this is that trees that differ in resistance must differ in their chemical composition somehow, however minor that difference is. and that we are able to detect or pick up those differences using infrared spectroscopy. So going back to the coastal live oak situation, we were able to identify trees in the field using classic lesion length phenotyping that were either resistant or susceptible to the pathogen. These are lesions that were produced after 9 months of incubation in the field, and we're talking about 20 centimeters here, but here we have up to 1 meter or more lesions in these standing trees with just 1 point inoculation on the stem. Turns out that these trees actually eventually died. So we actually killed them with those inoculations. So they were highly susceptible. And it turns out that we actually were able to classify them by analyzing their chemistry before the inoculation. Okay, so the considered chemistry. And so this paper came out back in 2014, all right, so it's a little bit of time. But essentially, we took extracts from the phloem. Fundamentally, they are phenolic extracts because we used methanol to do these extracts from the phloem, put them on one of these scanners, got a spectrum. The informative area or region is over here. This is mostly water. And then by doing statistical analysis, which is somewhat similar to PCA, but it's actually called SIMCA, which stands for Soft Independent Modeling of Class Analogy. We could actually classify resistant and susceptible trees both in PCA space but also in SIMCA space quite accurately. So here's how it works. You're supposed to use a set of trees to train the model, right? And then you're supposed to test the model using a testing population. We did not have a testing population, so we use what is called cross-validation, which is a statistical tool to obviate to that problem. We were able to have accurate classification of trees in the range of 80 to 90% accuracy. Okay, so that was quite great, I would say, because if you think about the fact that all this work was done in the field with all the ecological environmental noise that you have there, it was really fantastic outcome as far as we're concerned. Second example is European ash resistance to Aminocyphus fraxineus, which is ash dieback. This is work that was conducted by Katerina when she was a postdoc in my lab, published back in 2018. And I will just show you very quickly. I don't have time to go through the data in detail, but essentially what we showed is that if you take extracts, so let me say first, we used genets that were replicated across several gardens in Europe from Austria, Germany, Denmark, France, Lithuania, and Sweden. Okay, so we had the same genets planted all over Europe, essentially. And we were able to demonstrate that if you took extracts from the shoots or the twigs rather than the leaves, you could separate resistant and susceptible trees, right, irrespective of their location. So this technique was able to detect resistant and susceptible trees no matter where they were planted, okay, with a high level of accuracy again. The third case study is Norway spruce resistance to Heterobacterium adenosum. This was a collaboration with Fred Asiegbu, who I'm sure many of you know, in Finland. And again, we were able to demonstrate that we could classify what he called asymptomatic and symptomatic Norway spruce trees. Turns out that actually means resistant and susceptible trees in this study. I won't show you any data about this because I don't have enough time. So, take-home message here is that IR spectroscopy coupled with statistical modeling can be very powerful to phenotype trees for resistance and can be very rapid once you produce a model. Okay, so first you have to produce a model that's based on actual phenotypes, right? But once you have that model, you should be able to apply it to any unknown tree and be able to say, this is resistant, this is susceptible, without having to do any more inoculations. So, we were very successful using statistical tools like SIMCA and principal component analysis, essentially. But we knew that artificial intelligence approaches are actually even better at detecting signals in the morass of noise that you have with any kind of spectrum derived from natural extracts. So what is it? For those of you who are not familiar with it, really it is a decision-making process that results in pattern recognition. There's nothing really magic about it. It's not really intelligence the way we understand it. But it's very powerful. There's various kinds. For example, machine learning, which is the most commonly used, which includes support vector machine, which is what we use in our further studies. And deep learning, for example, convolutional neural networks. This is a bit more in the range of what you call, or you can imagine, as artificial intelligence than machine learning. Machine learning is fundamentally a very advanced statistical tool for pattern recognition. And so SVM, at its foundation, it's a computer algorithm that learns by example to assign labels to objects. This is a great article by chance, if you guys want to take a look at a great explanation for what support vector machine is. So the learning occurs by way of a training data set, and then once the best solution is found by way of SVM, it is tested on a testing population or data set from samples of unknown classification. So we applied that to actually this is case study number 4. I lost count there. So this is Austrian pine resistance to diplodiopenia or sapenia or sphaeropsis whatever you want to call it. A lot of you, I'm sure, are familiar with this tip blight disease of many two-needle pines. So this paper actually came out in 2020 in Agriculture and Urban Forestry, and again, Katharina Willer is involved. Gosh, she's everywhere, it's just amazing. She's all over the place. So here's diploidal tip blight. The way we did this, we had a population of open pollinated Austrian pines in the greenhouse. We had about 90 plants, and we inoculated all of them, and then we selected the most resistant and the most susceptible groups at the extremes of the distribution to actually build our model. Okay? So it turns out that the training model worked with an accuracy, of 93%, right? Well, you say, well, training model, that's all in good. It's a training model. What does the testing model tell you, or testing population tell you? Well, we didn't have one again because we didn't have enough trees to actually do that. Normally what you do, if you have a large enough population, you take about 70% of the population to do your training and about 30% to do your testing. But we didn't have that, so we used what is called cross-validation again. This is the result of a 5-fold cross-validation verification. And we got to about 67% accuracy, which is not great, but it's definitely much better than tossing a coin, which would be 50%, right? So if you want to identify resistant Austrian pines to this particular pathogen, this is one plausible way to do it. You may, by doing this, you may end up with some individuals that are actually very resistant, some that are not so resistant, but in general, you have a better chance of identifying trees without inoculations using this than if you tossed a coin. So machine learning can be a powerful tool to phenotype plants for resistance. It's also been used, we've used this on eucalyptus and the chrysoporthic canker system in South Africa. I don't have the data yet because right now we are finalizing the data collection and analysis. This was in collaboration with Sanushka Naidu at FABI in Pretoria, South Africa. So in conclusion, I'd like to state that FTIR spectroscopy coupled with machine learning is a viable screening tool for tree resistance to pathogens and possibly insects. And I would encourage any of you guys who work on phenotyping trees for resistance to consider this approach. And if you're interested, just get in touch with me and we can talk and so on. I would say, though, that even more appealing recently, we've been able to develop similar approaches using a handheld scanner to actually measure near-infrared reflectance approaches. And we've already demonstrated its sensitivity for the detection of diseases at the pre-symptomatic stage. So again, using NIR instead of FTIR, using a portable scanner. And with that, I will take any questions you may have.
Thank you, Rico. You were perfect timing. Good. All right, I would like to call back here on stage Raul and Raquel for taking question, and I'll see if we have any questions. I know the coffee break is waiting for us, but we can wait. I see Paul, you have questions? No.
Yes, this was a question about the metrojasmate treatment of the Norway spruce seedlings. Yeah. I've seen some much better here, even were negative. I mean, they were more susceptible. And still you had as I remember, pretty large sample size. So do you have any idea why it turned out so non-resistant?
You mean methyljasmonate causing a reduction in growth?
No, that's quite typical, but if I remember correctly, you didn't have much, I mean, they were more susceptible too, in terms of mortality?
In terms of mortality?
Yeah, didn't they, I thought they died, more of them died than the control even?
Former studies have shown sometimes an increase in mortality as well, but in this case, my opinion, I'm neither an expert on that, has to be with concentrations, right, timing, and these kind of things. Supposedly, this concentration should be a good one, but still is increasing mortality. Also, a second source of this variation is environmental. It's the environmental circumstances, right? Many of these trials has been done under controlled conditions, and these guys are in the field. So, yes, how we apply the methyljasmonate, the timing of application in spring is different maybe from autumn before wintering, and the proper circumstances in the field that actually are they are open to environment. I mean, for example, just thinking about methyljasmonate may change some physiological mechanisms, and under some drought maybe triggers less resistance to the drought, and this kind of cross behavior could be triggering this mortality, but as you've seen, is significant there, right? Protect the seedlings that are alive, but triggers mortality. Maybe a reduction in the concentration.
Yes, so maybe could just mention that I think there's already more than 100 papers on methyljasmonate and conifers. So I know Melissa Magre at my institute and Adriana and me planning a review paper and see, because there's 100 papers and there's maybe 50 different methods. So it's a bit chaotic, but it will be interesting to see. There are some patterns.
You mean methods of application, right?
Yeah, methyljasmonate application on conifers.
Probably many of the different papers, they have different protocols of application.
Yeah, every different, I mean, everything you can imagine, and some sort of crazy ones.
Yeah, for sure.
Any other question? Jeff, the question?
I wanted to ask also about the methyljasmonate. So first, I'd just like to compliment you on your presentation. I really enjoyed the way that you put into perspective all the different types of insect-plant interactions. I wonder, Do you think it's possible that maybe the application of methyl jasmonate might have induced a different volatile profile that may have been attractive to the weevils that may have resulted in the higher frequency of attack? Stress volatiles, something like that?
What is for sure is attracting the insect because increase the percent of damaged plants, right? Reduce the amount of wounds, but attracts more. Not sure it's because a change in the profile, maybe it's because the amount of the volatiles. If the plant induce defenses, these defenses are going to be volatiles, and maybe increase the volatiles. And as I told, the insect is attracted by some volatiles from the stumps, but maybe from the trees as well, right? So, yes, it's attracting, and maybe because the induction that, I mean, doesn't make a lot of sense, but here there's a very good chemical ecologist that I'm a geneticist, but they increase the defenses, and increasing defenses attract insects. It's amazing, but in bark beetles, some pheromones they use came from the defenses of the tree, so maybe probably here you have people that can answer much better than me. But what I believe, my opinion, is just this induction of defenses somehow is attracting the insects.
Okay, thank you for 3 really nice talks. Enrico, I have a question for you. Because machine learning, simple assays and stuff like that sounds fantastic, but you have relatively limited data sets for obvious reasons. How much do you think I would need, say, to build a reasonable model for Diplodia in natural settings in Sweden where you could make a quick screen in the nurseries or something? How much data would I need really to make a model which holds fully?
Yeah, I realized that I actually uploaded the wrong presentation, wrong version. At the end of my presentation that I wanted to upload, actually I wanted to say that I have a video on my departmental webpage that shows both the scanning procedure and the downstream data analysis with the support vector machine, if anybody's interested. How long it would take? So we haven't tried NIR on Austrian pine. I know that Karina is going to be talking about NIR on loblolly pine. So she will show you how it can be done on needles. So we can definitely do it, and you probably know I'll be in Sweden in next year, for 4 months, and I'll bring the scanner with me to work on ash dieback. So I'm sure we could try Diplodia resistance as well. It shouldn't take that long, really. I mean, once you do the training model and you test it with a smaller population, and you validate it. Once it's validated, you should be able to use it.
This is also a question for Enrico. It's about your method. You said this 67% of accuracy. This is yes, this is relatively okay. I was wondering whether the problem lies in the fact that you're trying to put in sort of resistant and susceptible something that is actually in reality a continuum of resistance. And if you have considered other tools than this, maybe taking into account the quantitative approach rather than this 1 and 0, and yeah, I'm sure you have done, so maybe you can talk a bit about that.
Yeah, so we take the extremes to build the training model, of course. But if every time you test it, it gives you an idea of the correct identification of the resistant one also, not just the overall model, but the accuracy to identify the resistant group or the susceptible group. So usually it's very high levels of accuracy for each of those two groups. So when you go and test an unknown tree, if it is resistant corresponding to that sort of distribution in your range, you should be able to identify it. I don't know if that makes sense. Yeah, we can talk more.
At least another. I'll go from the back to the front.
John had a question for a while.
Okay, thank you. I'm Jing from Denmark. I have a question for Enrico. So I was thinking you test this method on different tree species. Have you seen any common pattern? It's like I was thinking it's like common chemical compounds from different species.
So this technique is completely agnostic. It is not, I mean, if you really want to, you can start analyzing the kinds of band absorbances or reflectances that you get and make some suppositions about specific compounds. For example, in the FTIR work we did with Coast Live Oak, we could say that hydroxyl groups and also aldehyde, I think, groups were important, which kinds of makes you think that maybe it is the phenolics that we were identifying using HPLC. But it really is an agnostic tool where you don't care about what the source of the difference is. All you care about is the classification power. It's like, I can say, this is this, and this is this. I don't care about. So, another thing that was not in that presentation I uploaded, I wanted to make the point that for people who are interested in mechanisms of resistance, for example, this is a great screen tool, right? Because you can actually identify resistant and susceptible trees, and then you can compare them using molecular tools, biochemical tools, and all that kind of stuff. I don't think we are there yet. I don't think you're talking about remote sensing. Yeah, using, for example, hyperspectral cameras. Aside from the fact that hyperspectral cameras cost hundreds of thousands of dollars, you can fly them, you can put them on satellites, but the spectral resolution is not such, I don't think, that you can actually pick up these wave bands from up there. It's something we're interested in, we are pursuing, but right now I don't quite believe it.
Hello. Yeah. Great presentation, and it's a pretty simple question. Did you try interplanting the provincial genotypes that you found rather susceptible in a given province or resistance in other? Did you try interplanting the susceptible and resistance genotypes of individual provenances to see if that changes how they behave in response to infection?
If we try to do inoculations again with a susceptible and resistant, in which condition, sorry?
So interplanting them, like if you have seen one genotype acting resistance in a given provenance, is it purely provincial, the resistance? Like you said, the susceptibility varies according to provenances, right? Mm-hmm. So my question is it purely provincial? Have you tried taking that genotype from that province and planting it to a different province to see if it retains its resistance or does it lose it?
I think I don't understand the question, because you say
Yeah. So is resistance purely provincial? That's what I'm asking. Do you think the resistance for some of the genotypes that you tested from various provinces, and you say that there is variance in susceptibility, right?
Yeah.
Across provenances. I'm asking, is it purely provincial? Is it purely based on a location? Or if you interplant them, bring them to a different province, do they still retain their resistance behavior? I don't know how to explain.
I think Enrico was right. He's asking about if this is a genetic or environmental effect, but I think you are tested in a common environment.
Yeah. It's because. Yeah. Well, sorry. No. It's just because I was not understanding the question, because the essay we did was we employed seedlings from different origins, but we did it in a greenhouse condition. So all the genotypes, all the provenances were under the same environment with a randomized complete block design. So That's why I was not understanding the question.
And then maybe put them back outside the greenhouse in the respective provenances, or outside the respective provenances. Do they still behave the same as they behave in a controlled greenhouse environment?
We don't know. Well, one point is we cannot, at least in Galicia, we cannot do it in any essay outdoors because it's a quarantine pest. It's forbidden to try to see what happens outside. But we just did once the experiment, so we don't know if there is any genetic bioenvironment interaction. I think that's what you are asking. Is it going to have the same behavior? We don't know.
No, with
I'm sorry.
With traits. No partnership.
Yes, my question's for Enrico. I'm John Mackay from the University of Oxford. Infrared, long interest of mine. I was wondering about the sensitivity and the resolution power. So how much of a compound has to be present in a tissue for you to be able to pick up a signal? And then how much of a difference do you need between individuals to be able to differentiate them?
The second question I don't have an answer to. The first one, if we use extracts, you're talking about extracts?
No, it could be a whole tissue sample or a ground sample.
Yeah, so I can tell you, with extracts, usually we have used a ratio of 100 milligrams of fresh weight to 1 milliliter of methanol, okay, and then it's cleaned up through through a process until you get to actual reading it on the FTIR machine. So I don't know what the concentrations of different compounds are at that stage, but it's all standardized to the same biomass, fresh weight, okay? When it comes to NIR, I have absolutely no idea because we're just reading fresh material.
Yeah.
Yeah, so I didn't show you, I don't know if Carolina is going to show them, but I didn't show you the spectra of, for example, resistant and susceptible trees. They are basically identical to the naked eye, okay? But it's the machine learning tool that picks up the minute differences in amounts at those different wavebands.
And I think
so you need a certain number of replications, of course, to be able to pick it up.
There's a lot of literature. These techniques are used for detecting adulteration of foods. We actually brought it in from the food industry or pharmaceutical industry. So we have not tested in forest environments or tree, but I think you can find the answer to what you were questioning, like what's the difference among specific level of a compound that you want to search in the, I don't want this to be, in the food industry. There's a lot of publication there. So they search it, they use it to search like rancid oils in potato chips or like products that are not supposed to be there in butter. So I think the level, it's very low what they can detect, but the exact data you can find it in food industry publications, and there's a lot out there.
Yeah, in the food industry, in food technology, they can, for example, tell the difference between organic and conventional butter. I mean, we're talking about those levels. Butter.
Right, one last question from Luis. Oh, good. Oh, and then Raquel has a question.
Just yes or no, I think it is. You said that with any species, you try with this NIR method, and if you establish a model, then you can use it in that species. Yeah. But is it true that you can use any size of the plant, any age?
I think as long as you're using the same material for all your measurements, you can use any stage.
It doesn't matter if it's.
I don't have the objective answer because we have not tested different life stages, but you need to take your samples, of course, all at the same physiological stage for all the plants.
Yeah, but my question was more about if you test with NER and it works at 2 years old.
Mhm.
Is it going? Do you think is it going to work?
Be predictive for later stages? No, I don't know that. But if your own phenotyping technique allows you to make that extrapolation, for example, with inoculations, then I would say the classification with NIR is just as good.
Okay. Yeah. I like very much these three talks together because it's, in one side is 1,500 plants, field phenotyping, lot of effort for getting genetic information. And the other side is inoculation, but with destructive methods. You are killing the genetic material, and then somewhat a new step. No? So it's just working with this. A general discussion or general idea is that working with induced defenses is very complicated. I mean, it's fussy because inoculation produces many changes in the plant, in plant metabolism and plant physiology, and both because of the induced responses to increase the resistance, but as well a lot of changes coming from effectors, toxins of the pathogens that we cannot differentiate from those responses that are willing to or are designed to increase the resistance. So this kind of method is non-destructive methods. I think it's amazing, but I think maybe just wondering if we have to go to a next step, and this is just for, and we are going to miss a lot of information, but can we predict the resistance, studying the constitutive stage? So what is the limitation for that? What do you think about it? You presented some of the results, but maybe there is in some systems, in some pathosystems we can do it, in another not, depending on the value of the induced resistance for each. So it's just...
Yeah, I want to make it clear that this work is based on classification of plants at the constitutive stage, right? But that's key. If you want to do a non-destructive assay, you cannot induce the plant to avoid the inoculation. I mean, I do a lot of work on induced responses, but this is not it. So it counts. This technique counts on the fact that resistance-susceptible plants will be differently chemically at the constitutive level to start with.
And so it makes sense to make this evaluation after the inoculation or after the challenge.
No, because at that point you might as well do the inoculation to phenotype. Right?
Thank you.
Yeah.
Just one thing. Wouldn't it be good for detecting trees on the field that are affected by the disease, but is not still showing it?
Yeah, I have a whole talk on that. I gave a whole talk on that at the Lisbon.
But that's it.
Yeah, we do. We have that. We published a paper this year.
So,
yeah.
it works.
It works.
Okay.
Oh, yeah. At least on one system. Actually, no. On one forest system, which is American beech and beech leaf disease. But we've also demonstrated it in rice plants, where we can if you inoculate the base of the stem, we can tell if the plant is infected just one day later, just by looking at the spectrum from the leaf. So it's a systemic effect. And also in soybean, we can tell if the soybean is planted in a soil with soy cyst nematode by looking at the leaves. And we can actually quantify the nematodes in the soil.
So for that reason, it would be useful for practical management.
I'll give you the reference.
All right, this was a very nice discussion, but now let's call it a break and let's all go for coffee.
stress ball close.
Ready? Are you good to start? Okay, so with the poster session, we have 8 posters. I know there are only 7 listed there, but 1 was missing. We have 8 posters to go through, so please, whoever is speaking next. The last one is added at the end, so don't worry. Look at the list, whoever is speaking next, try to be kind of close by, and I will just introduce your first name. You can go ahead and say your institution, all the rest, otherwise we'll never finish. And you have 4 minutes, which are short, so when you're 1 minute left, I'm going to try to stand up and be sure that we move through swiftly. And with this, we'll start with Asier.
Okay, thank you. I am Asier Rodríguez Larrinaga. I am from the Misión Biológica Galicia. I will shortly here explain a little bit of the poster I have outside. So, growth, defense, and reproduction are 3 of the main dimensions of life history of plants, and they have been shown to be under physiological and evolutionary forces that causes some trade-offs among some of them. And for example, here are some of those forces, like both herbivores and climate, have direct effect on the availability of resources for reproduction. Climate also directly determines to a great extent photosynthetic production and water stress. And chemical defenses to minimize herbivore damage are in many cases carbonate compounds which impose a cost to the tree in terms of carbonate resources. So we wanted to explore the role of resource availability and herbivore damage on the defense, growth, and reproduction trade-off in this must-seeding species. And we wanted to know if the genotype had an important role in this response. So to that end, we did 2 parallel experiments with clonal ramets, grafted 81 grafted clonal ramets, just a fertilization experiment with just 1 treatment, fertilized versus non-fertilized plants, and then an experiment with 4 different herbivore simulation treatments. I show here a summary of the results, but I want you only to pay attention to these 2 columns. That's all. We did not find any role of genotype in the response to these treatments in none of the variables considered. And lastly, this is the wrap-up about the trade-off between reproduction, defense, and growth. I would try to explain it. It's like this is not a 3D graph, but a 2D graph. So it's due to its orientation, all the points where the effect size for reproduction, defense, and growth are equal, all of them lie on top of the origin. So the displacement we saw from the origin is just the direction, the imbalance created by the treatment. The imbalance created by the treatment on this 3-way trade-off. So we can see here, for example, that the fertilization treatment changes this balance in the same direction, and mechanical wounding, which in turn changes it in the in the opposite direction as clipping, which is quite surprising, at least for me. And the methyl jasmonate application just reduces a little bit of the reproduction function. So you can have more details in the poster outside, and if you have any other question. Thank you.
Thank you very much. Next we have Marshall.
Yeah, no, the problem is that I need to recover the streaming. And I am not sure how to do it now.
Wing it by mind. No, I'm kidding.
So you are the number 14? Okay.
Yes. Hi. My name is Márcio Navrowski from the Brazil. This work is photosynthetics in plants of Eucalyptus bentami under LED light after restriction of irrigation. We work in the forest improvement, principally in Brazil. The luminosity causes physiological and morphological changes in plants, and adaptation of the plants depends on the luminosity. And the LED light is important sources different these morphological and physiological. The objective of this work was the test different colors LED lamps in photosynthetic in seedlings under water stress. In Eucalyptus bentami is important species in South Brazil because in South Brazil is cold too. The methodology is this Eucalyptus bentami is water stress in this grow room under different lights in the treatments. Different lights were white, blue, red, and mixed blue and white. After 40 days in acclimatization in these seedlings, weigh the evaluation, and when and 2 days after was intervals net photosynthetic rate in the IRGA, infrared gas analysis. In the results, in the 0 hours, no difference in the treatments. After 24 hours, the blue lamps have and the red have a good results. And after 26 hours, the blue treatments have the height average, and after 2 days after, the blue treatment have a good results. And the conclusion, in general, seedling of Eucalyptus bentami under blue light have lower restriction in photosynthetics with the possible of resisting for a longer period of water stress. Is this?
Thank you, Marshall. Next is Pavel.
This one.
Okay, so I will try to briefly introduce you to our research on the bark beetle caused tree mortality in the High Tatra Mountains in Slovakia. So this is the data we have. It's the landscape scale bark beetle tree mortality, dead or living trees. Okay. So our research question was that with this data, we should look at if there is a common variable, a climatic one, which can explain the patterns of the tree mortality on the landscape scale. Is it working? Oh, sorry. Okay, now, okay. So we have the data on the right-hand side. It's the infestation map of the bark beetles. The red colors are the infested trees, and the green colors are the living green trees. So in order to make some analysis, we converted this map to, on the top, to zeros and ones. The dead trees were 1 and the green trees were zeros. And we used 3 climatic variables: maximum temperature sums, mean temperature sums, and potential radiation. All these variables were modeled for the period from April to October, because it is the flying period for the bark beetles as well the vegetation period for the trees. And then we used 3 different modeling techniques, like, I will not go to detail, but they should be able to cope with correlated variables. For example, the mean temperature sums and maximum temperature sums, they correlate with each other. And so these are the results. It's a bit tedious to read the left table. There is each year of the outbreak, each variable, and basically red color means a weak variable and blue color means important variable. So in order to explain the table on the left, I needed to come up with the table on the right. So that's much more easier. For each year there is one variable which showed us some importance. Now we do not want to say that maximum temperature and potential radiation are the only ones which explain tree mortality on the landscape scale, but we think they can act as a surrogate for some other processes, for example, tree physiology or bark beetle population dynamics. For example, the trees which were able to survive the bark beetle outbreak, maybe they were less stressed against heat and drought stress. So that's all. Thank you.
Thank you very much. Andreas is the next. Okay, you're very efficient, all of you. Congratulations.
Yeah. The one with no cable.
With the cable?
No, with no.
With no cable.
Yes, my name is Andreas. I work at Skogforsk in the south of Sweden. This orange plop. And my problem is, I'm an operational breeder, so my problem is to find good clones to incorporate in the breeding populations in south of Sweden. So I'm working with Norway spruce, and Norway spruce populations up to middle Sweden, more or less. And the experiment that has been done is to test whether it has been shown previously that anosonum and parvipurum is not correlated in defense. So we would like to try to see if there is an environmental issue in these resistance. So what we did was 3 different experiments. One is the greenhouse experiment at, I think it's in south of Sweden. There we inoculated with parvipurum or anosonum. And anosonum was inoculated with 4 different inoculates. And as you can see, there is no difference between parvipurum or anosonum in a constant environment in a greenhouse experiment. So you cannot see any difference between those 2 resistance in this is leash length, so the length of the within the bark, not in the sapwood. So that's the first experiment, and then we tried. There is the green dots. That's the southernmost seed orchard for South Sweden. And then you have the green or yellow spots. That's 3 different seed orchards in middle Sweden. And in the green ones, you have ramets that are connected, and the yellow ones, you have ramets, the same clone, connected. So if we look at the green ones, the next slide, you can see there is you just did a Pearson correlation between these experiments, different years, 2015 and 16. And this was an inoculation in the seed orchard, branches inoculated. And then we measured the length of the growth. And you can see it's only in 1 year we have, for this Runestenen and Gatebo, there's the same genotypes. So you have a small positive correlation between 2 years. And Lars Lund and Sörgärde, we also have a small positive correlation, but it's not really big actually. So if you look at the next slide, you can also see that the take-home message here is that there is a huge environmental disconnect between the greenhouse experiment. So we need to be better to test the trees in real life before we choose the clones in the breeding population. And my next step is to write an essay, to try to... My next approach is actually to test all the trees I have in my Norway spruce for this. So it will be an effort to select the best clones. It's about 500 trees I should select to select the correct one.
Thank you, Andreas. Yes, go ahead with whatever you prefer. Do you need the pointer there, though?
So where do I have to point this then again?
Probably the computer, I say.
This one?
I think it's going to work.
Yes, press.
Just test this. Yeah, doesn't work.
Wait, it's not working because I'm with the stream stuff. Now try it.
Okay, fair enough. Okay, cool. Well, hi again. My name is Romulus. I work for the Royal Botanic Gardens Kew under Professor Buggs. I'm a postdoc there. My work can be resumed in a single question. Is there any genetic basis for susceptibility to acute oak decline? Acute oak decline, for those who don't know, affects most of the southern area of the UK. You can see that it's very easily identifiable by the bleeds. It's a very bad tree. Not all of them have that many bleeds. And can kill trees very quickly, like in 4 to 6 years. So it's a very big problem there. We know that there are a beetle and bacteria involved in this disease. We have identified, other people have identified that at least 3 bacteria are involved in tissue necrosis, and also the beetle the larvae of the beetle can make these holes within the trunk, and then it causes problems with the transfer of nutrients. Both the bacteria and the beetle, they're natural. They occur in the trees, even the healthy trees, and the beetle is native, is not invasive. So, and it may be the case that the plants are just weak and they're exploiting the tree, right? But we don't have any information on genetic predisposition to AOD, and that's what my project is about. So if we find a genetic disposition, then it's good. Then we can identify the trees with good breeding values, and we can use those to rescue populations, do a breeding program. If we don't find any genetic basis, that's okay as well. It means that it's something environmentally related, so we can try and treat the tree microenvironment pretty much. So I don't think I have to speak this, but for those who don't know what a GWAS is, which is mostly what I'm doing, is just trying to identify variations in the genome that are associated with a phenotype. In my case, it's case control. Right? Either the plant is sick or it's not. And actually, if people have ideas to how to make this a more like quantitative measure, that would be great, because there are a number of bleeds, number of holes in the agrillas. So, for example, we have information on AOD status, the size of the tree, which is kind of, you can tell the age of the tree by this. the number of exit holes, which is when the larva leaves as a beetle, number of bleeds, stuff like that. But so far, we've been using just case control. We have sequenced genomes for like more than 1,000 trees, 20 coverage from all these sites in the UK. We have a publication, not mine, but Gabrielle's already did some structural analysis with part of this data. And we still have 400 trees to be sequenced this year. So just preliminary results. These things will change for sure. But, I've done some population structure. You can see that we only wanted pedunculate oak, but you can tell that some of the trees were unfortunately Quercus petrea, so the PCA got them pretty easily with the hybrids here. We have also PCAs for the different sites, the introgression that I mentioned before, and LD. And I did a preliminary GWAS, and I found none of this is significant, but it's kind of promising because a lot of these peaks, they're associated with genes that I've looked around 5,000, 10 kilobases around the SNPs, and they have some very interesting relations. Some of them are products that are used for insect repellents and adaptation to high south conditions, stuff like this. So that probably indicates that plants are just more tolerant to whatever's going on in the soil, are less likely to get IOD, for example. So that's what I had to say. That's the collaborators, and thanks DEFRA for the funding and all the institutions involved. Thank you.
Thank you, Romulo. Bea is next. So I think the pointer might be out.
Okay. Hello, everybody. I'm Beatrice Toglio. I'm a PhD student at SLU and Skogforsk, and I'm working on ash dieback and breeding for resistance. So European ash has suffered a large-scale decline in mortality due to the invasive pathogen Hymenococcus fraxineus. The pathogen is native from East Asia, where it's non-pathogenic on Asian ash. While in Europe, the impact of the disease has been devastating. However, there is evidence that a small proportion of ash population shows tolerance to this disease, and this low susceptibility to disease, it depends on their genetics. So breeding for resistance is the most preferred method to ensure European ash survival. However, traditional tree breeding practices are time-consuming and highly costly. So chemical fingerprints have been uniquely associated with resistant phenotypes and can be used as marker for selection and speed up breeding practices. So in this study, we aim to characterize, to determine the chemical composition in different Fraxinus species that have different evolutionary history and therefore different susceptibility to the pathogen. And also to identify chemical biomarkers associated with disease resistance. So in this study, we use ash material from a clonal trial that was established in 2016 in southern Sweden, and we sampled both leaves and phloem from putative tolerant and susceptible European ash genotypes and 4 different Asian ash species. Then samples have been grounded using liquid nitrogen, and metabolites have been extracted with methanol and added shaftosite. To determine the chemical composition, we use LCMS, so liquid chromatography coupled with mass spectrometry. I still don't have any result, but what we expect that there will be quantitative and qualitative differences between susceptible and tolerant European ash genotypes, and also with Asian ash species. Thank you.
Thank you, Bea. Next is myself. I'll bring myself there. Okay. All right, yeah, this one. I have two, and I don't remember which. Okay, so this is the project of my former PhD student, John DeSoto. Oh, and sorry again, I'm Katerina from the University of Georgia. And it's about the response of loblolly pine to inoculation with fungal associated to the southern pine beetle. And so southern pine beetle, that's what we work most in the south, and loblolly pine in terms of insect, is associated with 3 different fungi, 2 of which are mutualistic, so they're carried in the mycanja, they're not pathogen, and they are symbionts with they mutualist with the beetle, they provide food to the beetle larvae while they develop. And the third one is an antagonist, Ophiostoma minus. And it's antagonistic to the beetle mostly because this is all off. Okay, no matter what. It's ugly, but the content is there. It's antagonistic to the beetle mostly because it's antagonistic to the other 2 nutritional fungi. So basically, the enemy of my friend is my enemy, something like that. And we know that it's antagonistic to the fungi mostly because it outcompetes them. It is a better competitor both for primary and secondary resource acquisition. So it gets foods way quicker than the other 2 species that are slow and non-pathogenic. But we had the question, can this antagonistic relation that the Ophiostoma minus has with the beetle somehow be also due to the relation, going through the relation with the host plant? In other words, the pathogen is a pathogen. It will stimulate defense response in the host, but it's also co-evolved to withstand those defenses and would mostly catabolize them or somehow don't care. While the 2 non-pathogen fungi are not pathogenic. They usually do not stimulate much response because they grow in galleries, which is already kind of that tissue. And they're not co-evolved to sustain or metabolize those responses. So we did a classic inoculation experiment. We have 40 trees. We inoculated them with all the fungi. We waited 3 weeks, and then we went ahead and quantified phenolics and terpenoids. And what we found that indeed the phenolic profile in response to inoculation with Ophiostoma minus, where statistical difference, we use a multivariate approach compared to all the other inoculations, to the induction after the inoculation. Of course, we're looking at the induction. And this is only for phenolics, though. While for terpenoids, we found no difference. So this is interesting seeing how the different groups somehow behave in a different way. Now, this alone does not answer to our question. The next step will be to perform bioassays with the few compounds that we found were specifically associated with the differential response, but we didn't get a chance yet to get to that stage. So it's still a pending question. Is the relationship among those fungi somehow mediated by the tree response? And I don't know yet, but we're working on it. All right. And that's it for one of my students. We can go to the next student. Thanks. Okay, and so this is the work of Simone Liming, my other PhD student, and those of you who were in Lisbon might probably have met her, or hopefully you interacted with her. And so in this case, we were looking at the effect of manganese fertilization on the defense mechanisms, still of loblolly pine, because that's what we have in the South US, and that's the majority of my work. So why are we looking at manganese? Well, the whole idea of the study came after we were doing several surveys in different plots, and we found that many times the only difference that we were finding between affected sites and non-affected sites, or even within the same site within plots, was the level of manganese in the soil. And so we thought, why manganese is the only thing that is giving some response? And so we went in the literature and we found that, well, we know that manganese is fundamental for photosynthesis, for instance, but what we didn't know at the time, at least, is that it's very crucial for many enzymatic reactions that are within the metabolic pathway associated with production of secondary metabolites, so for response to pest and pathogen, and in particular in the PAL enzyme and in the production of lignin, stilbenes, and coumarins. And so we said, well, maybe trees that are lacking manganese somehow could be more susceptible. So we wanted to determine the influence of manganese availability on the production of defensive compounds. And we approached this with a big greenhouse study with 180 seedlings that we treated for 9 months with differential level of manganese fertilization, so the standard one, 1.8 gram per liter, half of it, because we didn't know if by taking off all the manganese we were killing the plants. and then 0 manganese at all for 9 months. Now, those plants come with some manganese in the roots when you buy them, but after 9 months, they will have outgrown any residual manganese. So 9 months after, and those are called Georgia giants, they grow like a lot. In fact, in 9 months, they were like pretty big. But they would have consumed any manganese residual. So at that point, we went ahead and we challenged them with two different fungi, pitch canker, which is a canker, a blue stem fungus, so a vascular disease, we mock inoculated or unwounded. And after 3 weeks, we went ahead and harvest completely the plant. We actually cut it in tiny little pieces and divided and collected all. So we were searching of all, those are all the actual type of tissue that we collected. We were looking at different parameters, physiological, just to check if manganese had an effect on the plant, and then the defense reaction. And well, we definitely created a manganese deficiency. We could see a very strong effect on the fluorescence, of course, and on needles and stem content. But in terms of defense reaction, we were not as happy as we were excited when we started this project. There was only a marginal effect in the lesion length, so that was already a little bit of a bummer. Terpenoids, we're still working, so we are processing the result. I don't have them yet. Phenolics, we did find an effect, and that was interesting because the effect was only in the induced response. So at constitutive, you see nothing, but plants that were lacking manganese were not able to mount an equivalent defense response compared to the other trees. Well, no effect at all on lignin and also resin ducts. We counted and quantified the area of all of them, but there was nothing there. So yes, cool for phenolics. We were hoping way more, but that's what we found. And if you're curious, there's a poster. Feel free to stop by and ask more. Thank you.
Okay. Getting ready. Okay. So the next speaker in this session, session 4, is Aziel Rodriguez Tarinaga. Is that okay? Okay. He'll be talking about predicting terpene profiles in the crown of pine trees using airborne hyperspectral imaging.
Sorry, getting stressed. No pressure. No, this one, isn't it? Yeah. But I need a bit more time. Because I need to do this here now. Where it is. Okay. Sorry. And then this. I hope it's working. David, it's working. Okay.
Okay. Here I am again. So I'm Mr. Rodriguez again from the Mission of Galicia. Yeah. It's on. Okay, so we'll talk on some work we've been doing to predict terpene profiles and terpene presence in the crown of pine trees with airborne hyperspectral imagery. During the last years, the advanced growth of the drone industry has been, revolutionizing the forest science and forest management, especially in parallel to a growing availability of sensors and lower weights of the sensors. So nowadays, the drones have already been used to estimate and measure structural variables such as canopy height or forest composition, some inventory variables. They have been used also to monitor dynamic processes and functional processes like fire recovery and forest decay rates. and they have been lately quite used to estimate, to measure, remotely measure some tree traits like nutrient contents, specific leaf area, and so on. As regards tree health, they have been already used to measure herbivore attack, tree disease, and abiotic stressors. We have here some examples. But mostly in this area, the focus has been in identifying the signs of tree illness, of tree weakness, which is supposed to be consequences of the attacking agent. But there has not been too much work on identifying the intrinsic traits of the tree in as regards defensive compounds or strategies. So for Enrico's work. And so we wanted to develop a workflow to remotely detect the defensive volatile compounds of Pinus pinaster canopy by combining the field work and measurement of defensive compounds in needles taken from the field with the measurements taken from the drone-borne hyperspectral sensor. Specifically, we wanted to measure and describe the variability of defensive compounds in these pines, and then explore how predictable these compounds, these concentrations were when working from the hyperspectral sensors, and how these models were, how predictable were as compared to some multiple response models. I mean, from models that considered the whole bunches of the different groups of compounds. And lately, we wanted to predict the possible profiles of these compounds that they were found in different trees in different areas from these sensors. So the work was carried out in Zaragoza, in Zamora province, and this we have here the three localities. One of them, here in Alcañices, we had 2 plots here, and the stands here were healthy stands where there were no problems. We had a second locality, second site with 2 plots where here we found clear signs of decay, but we don't know the cause yet. And then we had a third one. It was 4 plots here, and there were close to some stands of Pinus pinaster in the Portuguese part of the border, which they had been recently attacked by the Ips beetles. So we took 6 flights at each of the 8 plots along the year at an altitude of 140 meters, which gives a GSD of 5 centimeters per pixel. We use this drone, which is DJI Matrix 600 Pro, and this is the sensor, Nano Hyperspec, which has 272 bands, and so it covers a range from 400 to 1,000 nanometers. It covers most of the visual and near infrared range, but it doesn't cover the shortwave infrared, which has signal for many important biochemicals like this, among them the phenols. So at the same time of each flight, we took samples from 7 trees, representative trees per plot. We took samples from the crown needles, and we just brought them to the lab and analyzed for phenolics, tannins, monoterpenes, asciiterpenes, and diterpenes. The terpenes were individually quantified. And we analyzed the relationship between the concentration and the spectra by means of partial least square regression, simple models for single compound models. Then we made multi-response models that were just one model for the monoterpenes, one model for sesquiterpenes, and one model for diterpenes. And then for the terpene profiles, we did a discriminant PLSR, because this is a categorical variable. And the analysis of the variability of the diterpene concentration was made by the simple PCA, and we extracted the profiles from there. The analysis of the PLSR was done
mostly following the workflow proposed by Barnett et al. Last year, which means that we have to do some repeating modeling. So we split the sample size in just a fitting or calibrating sample size and a tested sample size that is left apart until the end. Then we split again this sample in a training sample and a cross-validation sample. We use this to build the model. We evaluate this model with this cross-validation sample, and we repeated these permutations 50 times. From this, we just estimated the optimal number of components for the final model, and the final model was then fit to the whole fitting sample and tested from the testing sample. So all the performance metrics I will show here comes from this step. That means they are prediction metrics, not training metrics. Okay, so we found a total of 14 monoterpenes, 27 sesquiterpenes, and 15 diterpenes. We only used 12, 21, and 9 for the analysis because we didn't have enough sample size for the remaining. Here you have the whole list. And I have summarized the results of the models, the quality of the model with colors, so that you have the darker the orange color, the better performance of the model. So that means the higher the square error and the lower the error. And the green one is the best of the 4 categories. We see that phenolics and tannins gave a moderate performance quality model, and for the terpenes there is a high variability. I mean the quality of the performance of the model depends mostly on the individual compound. When we fit their multi-response models, I mean there are just 3 models, one with all monoterpenes, one with all sesquiterpenes, and one with all diterpenes. What we found, we have here, this is the response, the result for the single models, and this is the result for the multiple models. What you see is that in general there is improvement in the quality of the models. Higher square errors and lower errors, and this is especially so for the diterpenes, for which we found some models with square errors larger than 60%. When we look at the variability of the composition of diterpenes, we found that two principal components was enough to explain 68 of the variants, so we work with those. And when we plot all the samples in these two space to the two principal components in this space, we see that there are 2 groups that can be actually completely separable. So we call them profile A to this one and profile V to this one. This profile is characterized, the A profile is characterized because of these huge concentrations of these 3 diterpenes, which is isopimeric acid and dihydroabietic acid and abietic acid. And the B profile is characterized mainly by the lack of these 3 peaks and some relatively larger concentration of these unknown diterpenes and this trans-geranyl. If we look at the distribution of these 2 profiles, there is quite a surprising picture also, because we found that the B profile is almost exclusive of Travazos. Travazos was the site that was near to Portugal. We had 4 plots there, and the A profile is common to all of them. Then we try to predict the profile of each tree. I forgot to say that the profile it was stable for all the trees. I mean, all the trees had, in the 6 moments we measured them, they had always the same profile, not just a tree changed just once. So it seems to be like a tree trait. And so when we predict this from the hyperspectral measures, we had a model with an accuracy of 90%, and it's quite balanced because specificity and sensitivity are also very similar. So the rate of misclassified samples for this model was of 10%, which I think is quite impressive for such a thing. So to sum up, the concentration of at least some of the compounds, of the terpene compounds in Pinus pinastra needles can be successfully detected from the hyperspectral sensor, small enough to be carried out in a drone. These models, this predictability can even be, I think that they can even be improved if we consider the shortwave infrared region, which we didn't. We also found that there are some stable profiles of diterpene composition and concentration that are different for different trees, and that these profiles can successfully be detected also from hyperspectral measures taken from the air. And that's it. Thank you very much. And if you have any questions.
Perfect timing, Azier. Now, once again, God, I can't, she just keeps coming up. It's Katerina, and she'll be talking about detecting fusiform rust resistance in loblolly pine using vibrational spectroscopy.
I know. I feel like I've been up and down all the time. Okay. Thanks, Enrico. It's Villari presentation, I think. Okay.
Is this one?
It's this one, yes. Perfect. Okay, so, well, Enrico already said the title, so I can go ahead. And I'll start with... It doesn't go.
I need some moment, please.
I'll wait. No worry. Just let me know whenever.
Rafa is under pressure, guys. Can you give him a round of applause?
There you go. It works now. Okay. All right. So once again, I'm presenting the work of my student. They came to the Lisbon meeting. I could not bring them all here as well. But so this is the work of Simone Limhing. It's one of her thesis chapters, and it's a collaboration with two of my colleagues from UGA, Kamal Gandhi and Christian Montes, and also two collaborators from NC State, North Carolina State University, Kit Payne, who by now has left and went somewhere else, but Trevor Walker is still there, and the Tree Improvement Program. So the Tree Improvement Program works mainly in improving loblolly. That's where we work in the South. And I'll just mention right away that it's funded half by the Forest Service Special Technology Development Program and by the Southern Pine Health Research Cooperative, which is an industry-based cooperative that I co-founded with my colleagues 4 years ago. Okay, so the study system, it's fusiform rust. It is caused by Cronartum quercum, forma speciale fusiforme, and it's particularly aggressive on slash and loblolly pine, which happen to be the most important pine species that we use in the South U.S. for plantation. And so it is considered the most costly disease in southern forest. And the damage is not only that it kills seedlings, which is already a big damage when you're in plantation, but it's the fact that when the infection occurs early on, you're going to have defect of the main stem, and so you're going to have shape defect and loss of growth. And if you think in plantations, that's a big problem because you're going to lose value of whatever you're producing. But luckily, this is one of those systems in which breeding for resistance has been very successful. So it is a success story already. It's well developed, fairly successful. I think one of the reasons is that they put a lot of money into it and they start working a long time ago because they've been working for a long, long time. That helps with success when they throw you a lot of money to work on something. But another reason is that the recognition and defense response is really a gene by gene, so it's qualitative. So it helps, even that it helps with selection. However, even if it's already a successful story, there's always room to making things even better. So to improve this selection procedure. So how does it work? And I'm talking to a crowd who probably knows it more than me. I'm more of a pathologist, but we start with a recruitment population, something that for some reason we think might have potential. and we take pollen out of it, and then we start having testing progeny test sites, in which we look for the development of galls. And we'll look by just literally looking at them. Now, I don't think it's going to be dressed that way, probably with boots and something else, but if you have galls, it means that the trees are susceptible and you discard them. If it don't, then it means that it's potentially resistant, and you start testing the candidate resistant line, which get tested again and again for many years. So it is working, it has worked very well, but it has some problem, and one of the main problem is that all is based on looking for galls. And so, first of all, the rust disease have almost a 2 years latency phase, so you might not see galls, but it doesn't mean that they're not there, they're just to be created yet. Also, it's a person looking, and we are all humans, we all do errors, especially tons of those little seedlings, so there's always room for errors. And also, the last thing is that it depends on the pressure of the disease on the area. So if you have a progeny test site in an area where in that year there's not a particular pressure of the disease, you're not going to have gall development, but the line is still susceptible. So it's very common to have escape susceptible, which then you keep propagating in the field, and you don't have the effect that you want. Now, still a very successful, I mean, we don't have many of those, but it's still something that we would like to get even better at it. So the objective of the study was to develop a protocol for the rapid phenotyping of loblolly resistant to fusiform rust using the vibrational spectroscopy approach. And so I'm glad that Enrico gave the talk earlier, so he took all the hard part out of me, and I can go straight to the results without going too much into the detail. But in case you were not listening this morning, I can kind of like recap it very quickly. So, and when I prepared this, I didn't know Enrico was talking just before me. Those are the density molecules that Enrico showed. So vibrational spectroscopy is based on the fact that it takes advantage of all these different vibrations and stretching. And so with those tools that can look at different wavelength, somehow you can capture all those movements. Now, as Enrico was saying, you're not going to be able to tell what compound is there. That is too much. But you're going to be able to have a signature that is uniquely associated with each different sample. So how do we use it? We use tools like this one. This is the one we use in this study. It's a handheld NIR, but it could be a benchtop FTIR or a Raman. There's several different approaches that you can take. You shine the beam through a sample that will have some chemistry. I mean, that's what we're looking at. And you're going to end up with a spectra. And that spectra per se doesn't tell you much, but when you have a lot of them, you can use them as fingerprint for a pattern. And then you use those patterns to build statistical model that will predict a different phenotype. So how do we, this is the theory, how do you transform it and translate it to an actual operational things? Well, in this case, since loblolly has been selected for a long time, we have availability of many different families. And so we selected 40 families, 20 resistant and 20 susceptible, all based on the breeding value. And we selected those in a way that they were not related at all. That's important. We don't want to be predicting their, like if they're family related or things like that. We want families that are completely not related one to the other. But being a system that has been worked on for a long time, it was relatively accessible to have 40 families not related one among the other. And then we searched 8 testing sites all across southern US. That's, for those of you who are familiar, we have Georgia, Alabama, and Florida. We selected 8 testing sites in which each of the family, in a way that each of the family was in at least 3 of the site, but we have site in which all of the family was always present, and 19 to 23 families per site. So not every family was every site, because that was impossible, but there was a very good coverage. And we did it in this way because the way that we want to predict is not anything related with the environment that they are in. We want to be predicting their genotype. So it's important to have this kind of geographical representation and same families replicated many times. So for data collection, my student picked up a car and basically drove all outside US, bringing the little portable instrument with her. And for every site and for every family, we collected 2 non-symptomatic trees. So that's important. Enrico was talking about. We need the constitutive chemistry. If they're induced, then they're going to have a different signature, and we're not interested in that. So it was challenging to find non-symptomatic trees that are of the susceptible family. So that was a lot of search, but we were able to find them all. And so she ended up sampling a total of 340 trees with that tool. And she collected both stem phloem, basically you cut off a little bit of phloem and you lay it flat on the sensor. Here below you cannot see it, but there's a little circular sensor where you have the beam shining up. And needles as well. And the way she did it was like by just putting them all together and bunching them together. And we saw that even though you might think there's a lot of variability, there was actually not. So it's a really solid method. But there is one problem that we're current with. This was all supposed to happen in November. I ordered the instrument in April. COVID, whatever, it took forever to get the instrument in the lab. And so we didn't receive until Christmas. It was Christmas, but it was, Christmas present, but a little bit late. So we were forced to sample in February, and that's just not ideal, and we knew that. Because in February, in South US, you have a transition from dormancy, and then they transition into putting flower ready. Flower for conifers, of course, this is kind of flower. And they were already like bagging the trees. And so we knew that we had a lot of chemical changes in that month, but for logistic, that's the only time that we were able to go. And so we had to basically do it, knowing that we were not sampling in the ideal time. This is coming and going. So we ended up with a lot of spectra. So Enrico was mentioning this is how it looked like. You have all the spectra, very homogeneous overall, considering that those are all across US and so many samples. But how do you approach them? And Enrico kind of talked about it. We use the pipeline developed by Anna Conrad, previous PhD student in Enrico's lab. And this is, with the pipeline, you basically remove the outlayers, but all is not something, it's all basically regulated by the pipeline itself. It transforms the spectra to second derivatives, and then you test different optimal variables from all the variables. You bin them, or you use random forest to select the one that are the best. And then once you have that selected, you use support vector machine, so this AE method, to classify the different plants. And as Enrico was saying, it's very important to consider how you partition your data set. Since we have so many trees, we were able to really divide it in training and testing. So we built our model only on the training trees, and we kept a 30% completely untouched, and we used that then to feed it and test. And even in the training, it got divided another time into training and validation, because even for the training, you have to know how good is your training. So you're going to have a value for the training, which is given by this, tested on this, and then you feed new naive plants that the model never saw, and you validate that with your testing sample set. And you can do that in a proper way only when you have enough sample. But we have 360 plants, so we had enough sample. We could do that. But before we went and did the pipeline, remember I said we sampled in February, and we knew that that was going to be a problem. So we just went ahead and wanted to do a PCA and just see how are those spectra classifying. And alas, we were right. There was difference in the due to the phenological stages. This was sampled a week before everything else. Then there was a week of nothing. Then we sampled all, we were driving across, it was not easy to organize, and we had to coordinate with all the different people in the different partners. Then we sampled a bunch of sites all together, and then 2 weeks passed because my student had exams and a conference, and then she was able to go back, and we have 2 sites that were almost 10 days after. And you could clearly see how this is kind of all by itself, and those 2 are all by themselves. It's kind of the timing. So we knew that we had a lot of phenological variation, and that's not ideal. And we were like, but we had to deal with it. And we thought, what we're going to do, are we excluding them? And we're only going to build our model on the good central ones? But we said, no, let's include it all, because in reality, when you're in the field, you might not know exactly in what phenological stage you are. So it's good to test it. And, well, it still worked. So we were pretty happy. Despite that phenological variation that we know it's there, we were able to build a model that it was 98% of accuracy within the same group, so that's the cheating one. But that's the real one, 72, with a new non-tested group of samples. So that's a true validation, and 72%, it's really good. We're really happy. Now, we were happy up to a certain point, though, because it was working, okay, it's not a line barrel. It was working on extremes only. So this is predicting extreme phenotypes. When we try to reintroduce intermediate phenotypes, the model was not holding that well. So we're happy up to a certain point. But we knew that we were working with data that had a lot of problems to start with. Now, what comes next? We know that we scan those plants with a portable tool, and so those portable tools are sensitive, but up to a certain point. They kind of give up sensitivity for convenience, because going in a field and being able to scan with no destructive anything, no processing, in 2 seconds you have the measurement, it's very, very convenient. So right now what we're doing is that we are analyzing the same sample. So Simone in the field, she was collecting and sending it out. And we're analyzing it with a way more sensitive benchtop FTIR to make sure that we get, maybe we might be more lucky and get a better result with that. How much do I have? 0 or 1 minute? 2 minutes, okay, good. So how do I envision using this? Because this is how you build a model, but like, what do you do with it? So the idea is that, you know, when you get a recruitment population, you already had, you know that 70% of what you're bringing, it's probably going to be susceptible, you're going to trash it. So you're wasting resources there. So if we could go and screen our recruitment population even before we start doing all the progeny tests, maybe we're going to have a better chance that the majority of what we bring in and start investing resources in it will end up being actually resistant. So it's already an improvement there. And again, it's improving something that was already working well. So looking at getting even better. And also, if we use our tool to screen the progeny test, maybe we would really be able to avoid any escape susceptible. And so that's how I envision using this tool. I would still probably do inoculation, but with a way higher rate of success down the road. So that's how our industry partners are very interested in using the tool. And with this, I'd like to thank you all for the attention. This is Simone with her pines. Those are actually the pine of the manganese study, not the one of the NIR, but this picture is pretty cool, I thought. And please feel free to write me or get in contact if you have any question that we are not able to answer today. Thank you.
Thank you, Katerina. And the last speaker of the day is Osalador, or how do you pronounce it? Osalador Sampson. He will be talking, he's from University of Alberta in Canada. He'll be talking about comparative profiling of lodgepole and jack pine responses to infection by Cronartium harknessii.
Hi everyone. Hi everyone. Is it? Oh. Ready. Hi everyone. My name is Samson Osadolo, and I'm from the University of Alberta in Canada. So today I'll be talking about part of my PhD project focused on understanding the molecular mechanism of pines resistance to Cronartium harknessii. I already talked a little about this in Lisbon. I know not very many of us are familiar with the interaction of this fungus with pine, so I'm just going to give a little recap on this. So Cronartium harknessii causes the disease called western gall rust, and this leads to gall formation on stems and the branches of plants. So if we compare Cronartium harknessii with a typical rust, usually they undergo 5 different spore stages on 2 different hosts. So they usually have, they alternate between 2 different hosts. For Cronartium harknessii, usually they complete the life cycle just in 1 host. So they, affect pines directly. So they just spread from pine to pine. And this makes them, spread more rapidly in their geographical range. Now they are found mainly in North America, in Canada, and in the United States. In Canada, the main hosts are lodgepole and jack pine. Here we see the distribution of jack pine, which is more towards the east, and lodgepole, which is more towards the west. Just in the area in Alberta, we have their hybrids. There's been reports from previous field studies that lodgepole pine resistance increases with proximity to the jack pine range. But we've also seen that even in distinct population of lodgepole pine where they lack jack pine ancestry, there's also evidence for quantitative resistance. We've also carried out genetic analysis of the fungus, and we found that there are 2 distinct populations of the fungus. We have the east population, which are co-localized with jack pine, we have the west population, which also co-localized with lodgepole pine. We tried to also verify the resistance of both species to the fungus. Here we looked at 2 different provenances of lodgepole, 2 from jackpine as well as the hybrid. And here we can see that lodgepole pine tend to be more susceptible to the fungus if we just focus on these yellow bars, which indicate the seedlings with galls. So we see that they show similar responses when we subject them to both the west inoculum as well as the east inoculum. So we are trying to examine, the molecular mechanism for this quantitative response, quantitative resistance in response to the fungus. So we think that the mechanism for quantitative resistance, between species is actually different from the mechanism for quantitative resistance within the species. So just, as a baby step towards examining this molecular mechanism, we want to test first the hypothesis that the genes associated with chromatomagnetite resistance will be expressed earlier or at greater levels in jack pine than in lodgepole pine. I mean, it's going to be more intense in jack pine when we compare that to lodgepole. So we know that plant defense responses is largely dependent on defense hormones such as salicylic acid, jasmonic acid, and ethylene. Usually this also depends on the type of pathogen that attacks the plant. So if the plant is attacked by biotrophs, usually we see more involvement of salicylic acid. If the plant is attacked by decotrophs, we see more of jasmonic acid and ethylene. And between the pathways regulated by both of these hormones, we have, we can have either an antagonistic crosstalk or synergistic crosstalk, but typically it's usually antagonistic. And these pathogens can, exploit this antagonistic crosstalk, to manipulate the host to thereby then, suppress the defense of the host plant. So we want to look at, gene component of each of these pathways to see how they differ between both species. We carried out an experiment looking at lodgepole and jackpine from 2 different provenances, both in Alberta, and they were genotyped to make sure that we have pure lodgepole and pure jackpine species. And they were inoculated at 8 weeks, and samples were collected at 1 and 3 weeks post-inoculation. We carried out RNA sequencing, and we also did some qRT-PCR just to examine the gene expression of these 2 species. So here are the pathways for the biotrophic defense signaling and also the necrotrophic defense signaling. In red are some of the gene components that are not just differentially expressed in response to the fungus, but genes that showed different pattern of expression when we compare lodgepole and jackpine. Now, what's very important here when we look at the biotrophic defense signaling pathway, we can see that the genes involved in salicylic acid biosynthesis are not differentially expressed, even though the markers for this pathway are expressed. They are upregulated. But what we see here is that the genes involved in jasmonic acid synthesis are differentially expressed. And this is very important because when we look at rust fungi in general, they are considered obligate biotrophs, and we expect to see normally salicylic acid responses. Here again, these are just heat maps to show some of the genes we looked at. Here we can see again the genes involved in the ICS pathway. The main gene component involved in that pathway not being differentially expressed. And then mainly what we see here also is that when we compare lodgepole and jackpine, we see more of an intense response overall, both for the genes involved in the biotrophic defense signaling pathway, as well as other markers for these pathways. For instance, the pathogenesis-related genes, some chitinases, as well as some terpenes. So to have some sort of validation of this data, we selected some few genes for qRT-PCR. So these are the genes we selected for qRT-PCR. And here we can see that when we compare the qRT-PCR data with the RNA-seq data, we see more of a consistent result. And this is very interesting because since we have high genetic variation at the provenance level, normally we expect some differences for some of the genes. Like for instance, if we have, let's say, more resistant individuals in in the QRT-PCR experiment, it's possible to see that probably some individuals are either way more susceptible and we can have differences when we compare the QRT-PCR and the RNA-seq data. But in this case, we see more of a consistent result when we compare both QRT-PCR and RNA-seq data in lodgepole and in jackpine. And what's key here is that, again, similar to what I showed earlier for the RNA-seq data only, we see, again, an intense response. All these genes are much more differentially expressed in jackpine compared to lodgepole pine. So we are still trying to look at more genes that are differentially expressed, probably just only upregulated in jackpine and perhaps downregulated in lodgepole or vice versa in these two species. We're also trying to look at different families in each of these species that show different levels of resistance to see how they also differ when we look at defense genes component or other genes outside the key defense pathways. So for now, when we've looked at, we compare different families with different level of resistance. So for now, we only have the hormone data for these different families. Here we see 4 families, so 2 families of jackpine and 2 families of lodgepole pine. JP2 is less resistant than JP1, while LPS1 is less resistant than, LPS1 is less resistant than LPR1. And so we looked at the responses of these families in terms of how much of cytokine do they produce, following inoculation with the fungus. And what we see is that there's really no change in the levels of salicylic acid when we compare, the mock and then those treated with the fungus. So across time points, we didn't see any pattern that indicates salicylic acid involvement. And if we compare among the families, again, we didn't see any indication of the involvement of salicylic acid. Interestingly, we see that there was an increased level of jasmonic acid in all the families, and at 3 and at 7 days post-inoculation. At 7 days post-inoculation, when we compare the most resistant jackpine and the most susceptible lodgepole pine, we can see a clear significant difference, again, which correlates with our RNA-seq data. So our reasoning for this is that Coranatum acnescens probably has different lifestyle in the disease cycle. So it's probably a hemibiotrophic pathogen, starts with a necrotrophic phase due to the production of jasmonic acid at the last stage of its life cycle. We also think that it's possible that perhaps this is just one of the anomalies where the host defense involves jasmonic acid, because we are considering already, if we look at rust fungi in general, that they are obligate biotrophs, and so we should always see salicylic acid responses. But in this case, we are thinking that probably this is just one of the rare cases where we have jasmonic acid being involved, in normal responses against biotrophs. And lastly, we also think that it's possible that the pathogen is inducing jasmonic acid levels to suppress, the salicylic acid mediated host defenses. So just to summarize what I've said so far, we see that the defense gene responses to Coranatum acnescens correlates with the quantitative resistance level we see in both species, and that our result from the hormone quantification shows that salicylic acid is not really a key player in this in the response to the fungus. And lastly, we think that Coranatum acnescens might be suppressing the responses, the biotropic response through the jasmonic acid mediated pathway. So this is where it ends. But first, lastly, I would like to thank some of my lab members that contributed to this project to the point I've gotten so far, as well as our collaborators and also our funding agencies. So this is what it ends. Thanks.
Thank you. Thank you very much. I realize that I might have called you by the last name instead of, what's your first name? Is it Samson?
Yeah.
Okay,
Yeah.
Sorry about that. Okay, so can we have all the speakers come back up? And so we can have a discussion? Well, 3 speakers, right? Yeah. Thank you. I have a microphone, so I'll ask the first question. Samson.
Yeah.
So your hypothesis at the end really looked like an effort to fit your model, your system, into what we learn from, what we know from Arabidopsis and other model plants.
Yeah, yeah.
Is it possible that those boxes that things are put in, like biotrophic SA, necrotrophic JA, may not work in trees?
May not work in trees? Well, we know that in eucalyptus and other angiosperm trees, we see more of the same response to Arabidopsis, but we've not seen that exactly in conifers, for instance, in pine. So, yes, it might be different from what we would see, let's say, in angiosperms, like in Arabidopsis, but we don't know yet. So that's what we are trying to verify or confirm. Yeah.
Well, sorry.
So this is also for Samson and your studies. So have you tried treating your jack pine and your lodgepole pine with sage osmanate or salicylate and look at the response to the Chronichium infection?
No. We didn't do that. We intentionally just wanted to see levels of these hormones without any sort of maybe priming or, because we've seen that, yes, in conifers, if you treat them with, there are cases where Norway spruce, for instance, have been probably infected with some endophytes, and then later when they treat them with, they were primed with some endophyte, and then later when they were infected with, let's say, some pathogens, they show some cytokine responses. But we don't want that. We want to see just in their natural state how do they respond in terms of their hormone responses. So,
because you could also imagine treating with inhibitors of either pathway.
So inhibitors of the salicylic acid pathway and
Salicylic acid production or jasmonate production.
Yeah, we've not done that yet, but I think it's something we would probably try in the future. Yeah. Yeah.
Any other questions? I don't know why I have two microphones.
For Kate, you have shown that there was a big influence of the timing of the sampling, no?
Yes.
In the phenotyping.
Mm-hmm.
What about the environment effect, the location? Because I haven't seen any effect.
Yeah, sorry, I haven't shown in that because, due to the presentation, but no, there was no effect of environment. So those sites that were all kind of grouping together was one in Florida, one in Alabama, one in Georgia, and so it was the timing was the only one that was creating any effect, but the location did not. And that's why we designed it in a way to kind of cover a vast area.
Okay, thank you.
Yeah.
I will add to that that's the same thing we found with European ash. Right, location did not have any effect, except for if you looked at the foliage, yes, but the twigs had no location effect.
In the ash, the location had a strong effect on the leaves, but not on the
but
flow.
not the twigs.
In here, we didn't find it, not even in the needles, honestly.
Yeah.
There was no effect of location, but there was a strong effect of timing and phenological stage. So
do you think that your accuracy would be higher if you were able to sample at a better time of the year and closer together?
I can hope.
Well, just ask.
Yes, I say yes. I don't know. It's like we haven't tested. Yeah, I think we could, but it's not tested, so it would be kind of speculation.
This is another question for you, Kate. I think I showed, when you showed this figure of the PCA summarizing and you talk about this sampling date effect. I think the proportion of variance explained by the two first components was extremely high, something like 98%. Does that mean that the spectra is something like redundant or something like that? I mean, with only two...
Yeah, so I don't remember on top of my head how much it was explained, so I trust what you
it
said.
was very high,
Yes.
I don't remember.
That was just a quick, that was just a quick looking at the data. We were not doing any pre-processing there. So when you do the actual analysis, we have all those, like the random forest selection will take care of the redundancy of spectra.
Mm-hmm.
So the quick PCR was really just a quick way for us to see, do we have an effect of timing, and are our data good enough? So I would not draw any conclusion based on that, because that's really quick and dirty with all the spectra all together. In the actual modeling, we take care of the potential collinearity or redundancy of too many variables. That's why we use the random forest selection of variables, which will take care of that. Now, that's a really good point. So the PCA was just a quick way for us to say, hey, let's just have a look, but it's not made in a way to take care of all the variables in a correct way. Thanks.
So how many wave bands did you use in the end? Sorry? How many wave bands did you use in the end for your model?
With the random forest, there's no, it doesn't tell exactly. The way that the pipeline tells you, it doesn't tell you which ones. I don't know exactly. Maybe Simone would know.
Yeah, I think so.
I'll ask her. Good question.
Thank you. A question to Katerina. So does the, like when during the day, which time to measure is the best, or doesn't matter? Like does the light intensity affect results? We were sampling from the morning to the afternoon, so it takes some time. And so we started in the morning and went ahead until 6 p.m. So in the same plot, we would sample throughout the day. I don't think it really matters, because you cover the light. When you measure the phloem, you're covering, and so there's no sunlight that goes through. And with the needles, we have to put enough needles that there's no sunlight going through anyway. So it shouldn't matter that much. But yeah, we were sampling throughout the day. It takes some time to sample all the 300 plants. Yeah.
I think that what she meant was lighting effects on the chemistry. So if you measure your spectra like at 9 in the morning, are they going to be different than if you measure them at 5 in the afternoon?
Yeah, that's what I meant. We don't know, because we were sampling throughout the day, and we haven't checked if there was any difference between the sample recorded in the morning and those in the afternoon. But what I'm saying is that, oh yeah, now I see what you meant. Yeah, no, we haven't checked. So I don't think so, because the model holds well, but we haven't checked for that specific interaction. Yeah.
Sorry. Thank you. So I also have a question for Essie here, because I really didn't, it was maybe me not being totally on top of everything, but as I understood it, you measured in the visible blue light the near infrared red band area with the drone. So were you measuring terpenes, or were you measuring sort of general vitality?
Sorry, I cannot hear you very well. So
were you measuring the terpenes, or were you measuring the general vitality of the trees? What was, because my understanding of red band is that it's sort of just tree being happy, but maybe I'm naive.
I don't understand you very well. I don't know what you mean. But I don't know what you mean, because I don't understand the loudspeaker, so I don't know if
she was wondering whether you were looking at the red band in your samples, and whether that really predicted the concentration of the terpenoids rather than other associated factors. But he did partial least square regression analysis.
Yeah,
right?
actually, I did not measure the red band. I measured 272 bands from the 400 to 1,000 nanometers, and I used all of them to construct to build the model. So I'm using all of them. Yeah. But what I think that some of the compounds probably have a clearer sign in the short-wave infrared region that we did not use, because the hyperspectral sensor did not get that region. But
those hyperspectral sensors that have a wider range, they are very expensive. Yeah. Also very heavy. I don't think
that's the point.
You can't really fly them
with
on
a drone. You don't have too much to select.
Actually, I think that it is just one of these in Spain or two, so I don't know. It's like...
Luis. Okay.
Yeah, just for Samsung, in general, is that you are in your talk you can see this main question that is under our research, which is that we are measuring changes in the plants, but many of the changes can be not responses of the plant to improve resistance. Many of the changes can be made by effectors, toxins, or whatever, any other compound that the pathogen puts on the plant for interfere the signaling. No? So, just wondering how maybe you thought about how can we silence the hormonal signaling of a plant, of a pine tree, for producing knockout phenotypes like in the mother plant. Maybe can we use maybe no effectors from another pathogens, maybe chemicals, maybe we are very far away from silencing or CRISP or whatever, any other DNA technology, but it's just figuring out that we have to put that in. It's a main question. Why, if the pathogen modifying the signaling or whatever? So you just let me know what you have.
I know, like, we have a PhD student that has just come to our lab who is also trying to look at the factors of chromatomagnesi, and I would say I don't really know the approach on how to use these factors to probably knock out some or probably silence or suppress some defensive components from the plants. But I know that if we're able to do that, there are probably some approaches we can borrow from, maybe studies on Arabidopsis, for instance, to see the effect in the defense response from the plant. So, yeah, I think it's something we'll be probably looking forward to in the future, but I personally don't know how to do that. Yeah.
Something that we talk about many times is, okay, maybe if we know that a known caterpillar or whatever, any pathogen or pest can modify, we can use those proteins from those animals for as a chemical, treatment. Okay, we can synthesize proteins and we can apply that no effector instead to go to silence the DNA. So anyway, I think we should put more effort in identifying proteins or whatever thing in the phloem moving around, and it's for maybe it's just a general question for
I've also had some great suggestions, like for instance in inhibitors, like just as some that I've mentioned today, as to which inhibitor that can be used to probably maybe suppress maybe like the jasmonic acid biosynthesis, for instance, and see how that would affect salicylic acid response. So I think I spoke to you this morning regarding some inhibitors I saw in this presentation. I think there are some of these chemicals we can use, like the DECA inhibitor, which you talked about this morning. So I think it's something we might try. Yeah.
And maybe the biological compounds are much more effective and much more active than the chemical like the DECA or whatever.
Yeah.
So any advance in identifying effectors from pathogens can be used for another field of research. Okay?
Yeah. Thanks.
Thank you.
Yeah.
I think to stay on time, I think we have time for one more, maybe quick question, if there is one. Hearing none, I think we can call in a day.
Yeah, I need to, okay, make an announcement. Thank you. Yeah, thank you, everybody. It was a very nice day. I think, at least, I think we save time. We enjoy very much the discussions, and thank you very much for contributing to that discussion. Just for logistics, now we have a 6:30, Maria? 6:30, we have an offer from the Pontevedra Council for a guided visit, but You know, the forecast is not very good. It's not cold, but it's rainy, so maybe any of you can join the visit. And there is an option as well that they can move the visit for tomorrow. They call us now and say, okay, it's raining a little bit, so if you arrange, we can move the visit for tomorrow. So just in case, let me know who of you are willing to join the visit to Pontevedra. I think so. I think it's 45 minutes or something like that, no, Maria? 1 hour. Please move your hands for. Yeah, today. Anyway, today or tomorrow. Yeah. So. Yeah, tomorrow. That was tomorrow. We have a wine tasting in a bar over here. So. That is the point. So, for today was. I think tomorrow is even more rain. That is tomorrow. So the forecast for tomorrow is not very good. So that was the reason that we put the wine tasting, because it's not very nice to be outside. So I will say that's to do the visit today. I think so. Yeah, can we keep the visit for today? Okay. The wine tasting by yourself. Yeah. So Maria, it's 6:30. Just here in the Liceo Casino. Okay, just in the main door. Okay, and then at 8 we should be little quarter to 8